Hello, everybody, and welcome back to Creation Myths. We are live tonight. I've got a really fun show for everybody tonight. We're going to do a deep dive into a phylogenetics paper. We're going to crawl through the methods. It's going to be great. I'm so excited. Before I go any further, even introducing my guest, I just need to make sure that everything looks good on the YouTube end. It does. Chat, as always, please just let me know if there's any audio or video issues as we go, because I will, of course, have the chat window open for the duration of this experience that you're sharing with us tonight. So before we get any further, let me just take a moment to introduce my guest. Erica Gutsa-Gibbon is here. You all know Erica Gutsa-Gibbon. If for some reason you are here and you are not already subscribed to Erica, she's linked down below. You should absolutely go do that. Erica, how are you doing? And just tell us about what you have going on.
Man, I'm so excited to be here. I'm excited to blow off some steam on this one because I don't even know how to describe my feelings towards the interactions that you and I and also Professor Dave have been having with Dr. Tour and Dr. Stadler, which is the topic of today's video. And it's like, there are people out there who are going to be like, why do we need this video, a deep dive on like the phylogenetics of several different papers going in gross technical detail for a video that's effectively on YouTube? And the reason is because they forced our hands. They have forced us to sit down and walk step by step, bit by bit through this. I've learned stuff about phylogenetics over the past 2 weeks that, I was like dusting off neurons from undergrad with some of this stuff. It's welcome. It's good. It's going to be great. You guys are going to learn a lot. I certainly did going back and forth with Dan on this. And I'm probably going to have more questions still. But the long and short of it is, as usual, we've got some creationists who are misrepresenting or misunderstanding, or both, depending on the person in question, a paper that is quite harmful to them. And so we're going to dig into the paper and see what it says.
Yeah. And I just want to second something that you said about dusting off neurons. So my lab in graduate school, which I graduated 10 years ago, and we did viral phylogenetics. So my kind of contribution to that work, like one of the things I did that was specifically related, I had my own stuff, but I also related to that. One of my projects was documenting the mutation spectra of viruses. So like to generate the parameters for the substitution matrix, right? Like A to T versus T to A. Right. How do you score those when you're doing the phylogenetics? So I was kind of setting the parameters upstream, and then a bunch of my colleagues were actually doing the phylogenetics. So I became pretty familiar with a bunch of the stuff and some of the software. I love reading papers and being like, I know what that is, when it's, it's some program or whatever. It's like, I remember that back in the day. And so this stuff was familiar to me, but also it's applied in a different way from how, we did it. So I had to, go back and wrap my head around it. It was a really good experience. It feels good to kind of flex those muscles a bit. It was really nice doing this. So let me tell you all how tonight is going to go. We are going to do this in kind of 2 parts, okay? First thing we have to do is do a deep dive into a 2016 paper that I'm going to pull up on screen in just a minute. It's Baum et al. Statistical Evidence for Common Ancestry Application to Primates. We're going to go through this paper focusing on the methods. We want to understand what they did and how they did it in order to reach the conclusions that they did. We're going to really take our time with this, including going to a couple of the references in that paper that further explain the methods. Because my goal here, and this might be ambitious, because this stuff is complicated as hell if you don't do it for your job. It's really hard and not intuitive to wrap your head around. But we're going to try really hard to get to the point where you have a general understanding of this paper without any background except for basic high school biology. That's what we're going for here. But then we have a second thing we're going to do, because as many of you know, Dr. Rob Stadler recently released a video on Dr. James Tour's YouTube channel about this paper and us. So we're going to just take a stroll through that video and pull out some highlights from Dr. Stadler and comment on those. he made some of the same errors that basically every creationist who has ever talked about this paper has made. There was like some people on Discovery Institute, if you go back like a bunch of years, made basically the same errors that Dr. Stadler is making now.
Emily Reed did this a couple years ago.
It was Emily Reed. Yep, exactly. So what we're going to do is we're going to do this deep dive through the paper, and then we're going to look at what Stadler's critique is, and we're going to see how basically he is just fundamentally, I think, misunderstanding the words he is reading in terms of how they apply to the methodology in the paper. So I want to be really clear about this. The first part, the deep dive into the paper itself, we are playing this completely straight. Like we can have this conversation with no reference to creationism or creationists at all. It's just let's understand how this paper works. It's complicated paper. The methods are like very specific. So we're just going to walk through it. And I do want to note that we invited Dr. Stadler to join us for this conversation. He obviously declined. We did make clear that we will, accommodate your schedule. We'll make it work if you want to be here. And the three of us can just waltz through this paper together, working through the methods. I think it's unfortunate that he's not here because I think working through the paper together with him would be a really productive thing to do. Because then we can shout our understandings and lack of understandings altogether. And I think that would be good.
I understand the hesitation because, like, I just did this with the James Tour thing where James Tour wanted to have a conversation with me on abiogenesis. And, my first question was like, why would you want to have a conversation about abiogenesis with me who doesn't know anything about chemistry? The last biochem course that I took was an undergrad many moons ago. So I understand the trepidation on Rob's part because it's like, okay, you would be coming into a room as somebody who's a non-evolutionary biologist with an evolutionary biologist and, a PhD candidate in an adjacent field, biological anthropology. So it's like, I do understand the trepidation, but like, I wish it had happened. And what I would like to say is that I'll speak for myself, but I'm going to speak, actually, scratch that. I'm going to speak for me and Dan, because I know he would agree to this. We'll sit down with Rob one-on-one or whatever as a trio, like in a private conversation, and explain it if you would like, because it's like this, there is an element of YouTube that is about like, dunking and scoring internet points. And like, I love busting creationism as much as the next person, probably way more than your average person. But there's also an element of this that's like, it's important to understand what the paper is saying. It took me a while to figure out what this paper was saying too. So it's complicated. It's complicated. And so it's like, I would like to extend that invitation because this isn't in his background. And I think we could at least move forward on this specific subject if everybody's on the same page there. So that invitation is out there, Rob. I know you're going to watch this eventually. Email us, one of us.
Seriously, I co-sign that. Yes, I think that would be productive as well. Good. Okay, so folks, let's do it. The reason you all are here to just go into excruciating detail on a paper on phylogenetics. Okay, so this paper is Statistical Evidence for Common Ancestry, Application to Primates by David A. Baum et al. This was in 2016. And this is just a really cool paper. Like this paper, they describe in the introduction, like kind of how this paper came about. And it basically came out of what, like essentially a graduate seminar of like, how can we actually test the hypothesis of common ancestry, right? What can we actually do to evaluate the validity or the strength of that hypothesis relative to alternatives? And there's a really interesting thing here, just a teeny little thing that had nothing to do with creationism. It was basically a philosophy of science question of like, here's a tricky thing to actually evaluate. How can we do it? And so they actually go through in this paper a bunch of different tests of common ancestry versus separate ancestry. And we are going to focus on one of them out of like a half dozen. There's a bunch. We're just going to look at the molecular phylogenetics, and essentially genomic phylogenetics is what we're doing here. We're not going to do any of the other stuff. We're just going to ignore that. So,
for the record, though, we only ever see creationists have an issue with the genetics portion of this, which is a little bit interesting because this includes the fossil record and morphology, and it includes biogeography, two other in and of themselves, really three, if we've got fossil record, comparative morphology of extant species, and biogeography, three separate additional fields that, as you will see, because I think we're planning on pulling up the table that shows some of our values for several different categories, but if not, we can pull it up at some point. All of these body creationism separately in and of themselves. But for whatever reason, they never want to talk about the other ones. Maybe it's because we're the ones always bringing it up, and it's always reactive on their part. And so they talk about whatever we've pointed to, but I digress.
So one thing I want to just start with this paper is that, like, the reason this always comes up, and it's really funny that this came up when we did that video with Professor Dave. Now, the original one was a couple months ago at this point, the beginning of this whole saga, because we in particular love to bring up, like, when people talk about what is the best evidence for evolution, what's the strongest evidence for common ancestry, our answers are the same at this point, because we've talked about this so much, that it's nested hierarchies across all parts of the genome. Right. You get this nested hierarchical pattern, and there's no way to generate that pattern with separate starting points, regardless of what those starting points look like. You can't get that pattern, right? So we talk about this all the time. This just comes up when you're arguing with creationists. And this paper is basically like a formalized test of that explanation, right? So they're really digging into that and showing mathematically and computationally that what we've been talking about is the case. And for anybody kind of wondering how my thinking on this started, it actually started with the question of how can you document the kind boundaries, right? In a separate ancestry model, how can you document the separate kinds? And you've got lots of organizations like the New Creation people are all over this, trying to find like morphological matrices to draw the lines between the kinds. You don't need that. You do it genomically, because you build a nested hierarchy, and at some point, if there are actually separate kinds, the nested hierarchy stops working. It breaks down. You lose the neat clades, and it just becomes a big jumbled mess if they were actually separately created, right? And so you take that answer to how do you find separate kinds and flip it around, you don't. That's the strongest evidence for common ancestry right there, right? So that's kind of the lead into why this paper has the importance that it does in this overall conversation.
Just to be clear for anybody out there who might not be, aware of the consistency of this, right, is that it's like the way that we get a family tree within a species or within breeds within a species. So like dog breeds, or all humans, or all cat breeds, or all equids, or all felids, or whatever, the same exact methods that you use to get those family level or genus level or species level or population level family trees, the same methods that create those trees that everybody agrees with, joins all of these trees together in a tree of life. There exists no stop sign, no gigantic neon light that says, hey, our genetics are breaking down here, or our morphology-based phylogenies are breaking down here. That doesn't seem to exist, and we've looked. So right. That's the critical part, is that it's like, where's the line drawn, and why is it drawn there for these created kinds? And if the line can't be drawn, which I think that's where we're going to be moving today, what does that say about creationism? Where must they move?
Right. What does that mean for separate ancestry as a hypothesis, right? So just very briefly, I want to highlight a single line in the abstract here. And folks, I know that the text may be small on your screen. I apologize. We're going to have to, show the width of the page. So we're going to do our best to make sure you can read it, but I hope everybody can read this. I just want to point out this line. This is the big takeaway right here in the abstract. We overwhelmingly rejected both species and family separate ancestry with infinitesimal p-values. And we'll see those p-values in the table later on. So the starting point for this is they did these various tests, one of which we will talk about, evaluating the separate ancestry and common ancestry hypotheses, and they overwhelmingly reject the separate ancestry hypotheses. That's kind of our starting point here. So let's go down to the methods. Crack your knuckles, folks. We're going to the methods section. So we're going to get down here into, we're not going to worry about this. We're going to look into the materials and methods. So in a scientific paper, the way this works, your abstract is just a little paragraph of what did we do? Why did we do it? What did we find? Just boom, 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 real quick.
TLDR.
Yeah. Then you have your intro, which is all the background information you need to understand what's coming. If there's vocabulary that you need to define, whatever, that's going to be in the introduction. It's also going to be a brief overview of what we know in the field, what's been done up to this point related to the question the paper is asking. We're going to skip all that stuff. We kind of talked about that. So we're going to go to the methods section, which is what did we do? And the key thing with the methods section when you write a paper is it has to be specific enough that somebody could take those methods and recreate what you did, right? So they'll explain what they did and either have the details right there, or they will reference another paper that has the specific details, or they will have a supplement that has the specific details. And for phylogenetics papers in particular, very frequently you'll have a supplement that'll have like the parameters for the programs they used. And a lot of times it'll be like on GitHub and you can just kind of like load it up and do it yourself. So
sometimes they'll put the code itself, like for the UNL stuff, which we both just recently dipped a toe in, right? It's like they've got the code itself written out so that you can run these scripts or whatever and replicate whatever it is that they've done. Because as we all know, replicability, that's a really important part about running any kind of experiment if you want your results to hold any water.
Exactly. So the hypotheses we're comparing here are going to be common ancestry. So all the organisms in question share a single common ancestor. We're going to have species separate ancestry, where every species has an independent origin. And then we're going to have family separate ancestry, which is an interesting design choice, let's say, considering that most creationists approximate kind with family, right? So that's convenient for us. I don't know if that was coincidental or not, but it sure is convenient that it gives us an apples to apples comparison.
Given that this was in 2016, when baraminology was sort of swelling back to the public consciousness, that the shame of Dover had worn off and the creationists were starting to kind of put their stuff out there a little bit again. I don't think it's that surprising that they went with family level, because they probably just sat down and Googled, like, what do creationists think about nested hierarchies? And boom, it takes you right to baraminology, because it's completely indefensible to hold to any kind of species level separate ancestry, because we watch speciation happen. We have seen new species form in the lab and in the wild. This is non-controversial. So family level separate ancestry is really what we're interested in here with regard to testing like baraminology or the forest of life. This idea that God in the Garden of Eden 6,000 years ago, or if you're a Discovery Institute person, an indeterminate amount of time ago, creates these archetypal cats, the felids, that diversify and form all of the members of Felidae, and archetypal dogs that create all of Canidae. And it's like that for virtually every family, except when it isn't, which is when it changes to being, at the class level or at the order level or at the genus level or whatever. And this is especially the case when it comes to humans, who are just cherry picked out of the entire situation, so that they cannot be shown to lump with other apes, because that would just be that would be a bridge too far. So it's also quite lucky, right, that not only are they testing family level separate ancestry, but they're doing it with primates, which is uniquely damning to creationism.
It is convenient. Let's say it is very useful for what we do in the applications where we encounter creationists. Yeah.
Right.
So we're going to focus, like we said a little while ago, we are going to focus on just the first test that they did, which here in the methods, it's agreement within a single matrix is how they describe it. And this is basically molecular phylogenetics. So we're looking at a data set of gene sequences, and there's a bunch of different components to that. We're not going to go into it, but you can go into the methods if you want. They describe the Perlman data set and what that entails. But it's molecular phylogenetics. We're comparing various genome sequences across lots of taxa. And the way we're going to evaluate the support for a phylogeny, it's right here. I'm going to make this a little bit bigger, which might mess up this. I can't really make it much bigger. Zoom in. There we go. I'm going to have to just zoom around a little bit. But this is the key line right here. Parsimony as the optimality criterion, and hence tree length as a measure of tree-like structure. So that's the first line we have to really parse here for people who are not familiar with phylogenetics. Because basically what this sentence is telling us is how we're telling us if a given phylogeny is well supported or not. Is there a strong phylogenetic structure, or is there a weak phylogenetic structure in those underlying data? So maximum parsimony, everybody. The key here is going from common ancestor to all the descendants as necessary, wherever you draw those lines. The thing with parsimony is to minimize the number of evolutionary changes that have to occur over that tree, on all the branches of that tree. So, like, for example, if you have a bunch of taxa and some of them have a G at a specific site and some of them have an A at a specific site, and they're otherwise all identical, you're going to make one side of that tree, everyone's got the G, the other side, everyone together has the A, because that means you only need one mutation to explain all those differences between those taxa, right? A G mutates to an A one time, and then that explains all the differences. Whereas if you mix and match them, you need to have multiple instances of that mutation happening across your different groups. That's a less parsimonious explanation, right? So we want to minimize the number of changes. And another way to think of this is right here, tree length, because the branches of a phylogenetic tree are typically scaled to represent the number of changes to go from the node to either the next node or the taxon at the tip of the branch, right? So when we're talking about making an optimal tree, which is something we're going to be talking a lot about here, we're talking about trees that have the shortest possible length, which is to say the fewest evolutionary changes to go from ancestor to descendants. That's what we mean by maximum parsimony.
It's almost like an Occam's razor for an evolutionary tree, for a phylogenetic tree. It's like what's the easiest way to get to point A to from point A to point B, right? Yeah. So it's like you want the fewest amount of steps. That's going to be like your most parsimonious, geez, tree. Now, Dan, we talked about this beforehand, but like this has been a fun foray for me because I study gross morphology, so I haven't touched phylogenetics outside of like phylogenetic corrections, like to account for other statistical tests, and like when I'm running my stats in like R or whatever. It's like I haven't done phylogeny in a long time, so it was fun to get to sit down and read some of this, and like Dan explained a lot of this stuff to me beforehand. But I'm here in part to act as the audience surrogate and ask some pointed questions to make sure that we're all on the same page. So Dan, let me ask you, does parsimony work the same way at higher taxonomic levels as it does at lower taxonomic levels? Would parsimony work the same way if I were to create a phylogenetic tree for dog breeds or human populations as it would for, say, primates as an order?
Yeah, you can apply it at any scale, basically. And it's funny you ask it that specific way because, like I mentioned, my history in graduate school, we did viral phylogenetics. A lot of that was looking at strains of the same virus. Like, for example, I had a colleague that was looking at strains of a virus in a particular outbreak of a plant pathogen virus it was a Gemini virus. So that's all the same species, but they're looking at the history of that virus and then plotting it, spatially, kind of tracking that outbreak of that virus as it transmitted through various regions in their crops. And so, yeah, you can do this at any level. It's independent of the scale. It's the logic that matters, right? The idea here, like you said, is this is Occam's razor, right? You're minimizing the number of mutations that are required to get from point A to point B, where you have the extant taxa, right? Whether that's strains of a virus, all of the same species, or a bunch of different primate species, or whatever. You have the extant taxa, and now we have to work backwards and find what that most parsimonious route is back to that common ancestor.
Golly, that sounds like observable science to me.
That is observable because you're not making, we'll talk about this a lot, but you're not making any assumptions about what those nodes, those common ancestors in your tree represent, right? You're not characterizing those a priori. You're letting the data kind of guide you to what the best pathway is back in time, right? You're not saying we're going to start with this and we're going to end with these, and what does that pathway look like?
And to beat a dead horse and remind everybody, we're doing this for primates in this paper, but the methods are exactly the same if you were to do it for populations or dog breeds or cat breeds or horses or whatever. The logic works the same way. No necessary evolutionary assumptions required, at least that I am aware of.
The only assumption is that, like, as you said earlier, like genetics works, right?
Works
inheritance
the same way
works.
now as it did in the past. Yep.
It works, right? At no point do the fundamental mechanisms of inheritance stop operating the way we can directly observe and document them operating, right? Given those parameters, right? This process works going back in time. We can make hypotheses about what that evolutionary history looks like based on the standard of maximum parsimony. So that's the standard we're using, folks. I will say there are other standards you can use. There's maximum likelihood, for example, is one that was mentioned in this paper. And we're not going to go into it. It's just maximum likelihood, rather than optimizing for the fewest changes, optimizes for the most likely series of changes, which may not be the same thing if, for example, a certain type of mutation is significantly more likely than another. Which does happen sometimes, so there is a robust case to be made for maximum likelihood methods. They are significantly more computationally demanding and usually return results that are pretty much the same. But there are other ways of doing this. I am just taking a moment to remind people that if people are being disruptive in the chat, mods should feel free to block them, because this is a science night, not an argue with Dan night. So feel free, mods, knock yourselves out.
Or if you want to set up a talk with Dan or I, we both have emails, and I know one of us answers them regularly.
And it's you.
I promise it's not.
Okay, folks, we both have real jobs and obligations that demand, lots of time. Sorry if I am slow answering emails.
We're doing our best.
We're doing our best. Okay, so let's stick with this paragraph because now we've established what the standard is. So how are we going to compare our different hypotheses? How are we going to evaluate if a certain explanation is good or not good? Well, now it's time to get into one of the linchpins of the entire thing. This line right here, the PTP test, right? We use the PTP test. What in the world is a PTP test? So this is really interesting. It's the permutation tail probability test. And this is adapted from two earlier studies here. So it's Archie 1989 and Faith and Cranston 1991. And in order to understand the comparisons we're doing here, we have to dig in to what a PTP test is. So I'm going to put this paper down. We're going to switch first to Archie 1989, and then we're going to pull up Faith and Cranston 91, and then we're going to come back to this with a better understanding of what we're doing here, because these other two papers, I think, do a really good job of clarifying exactly what it is. So let me remove this. I need to
pivot,
stop my screen. Then I need to share my screen again, pull up the different paper. Now we're going to do Archie 1989. Here we are. That's Faith. I have to pull it up on my screen. There's Archie. And now we can add this.
Sit back and relax while Dan.
I have so many things on my screen that I'm going to be taking up and putting down. It's just it shows like this. What I need a third monitor is what I'm figuring out here. Okay. So this is called a randomization test for phylogenetic information in systemic data. This paper is so old that when you download the PDF, it's not read as text, so you can't even highlight the text. I just had to block things that I thought were important.
And with these, Control F is variable too. Like some of these stand ones, sometimes it'll work, sometimes it doesn't, and then you're like, well, I guess I just have to comb through this by hand.
So let's talk about this paper. And I'm not going to read from this, but I'm going to have it on screen. And if people want to follow along, you can. We also have to just talk about some vocabulary here, which is characters and character states. Okay, so a character is a describable trait of an organism. It can be morphological, right? Hair color is a character. Height is a character. A character state is what you actually have for that character, right? Red hair, 5'10", whatever it is, right? Those are character states. When we're using molecular data, the letters in your genome are the characters, right? Each letter that you have is a character, and which nucleotide base you have, A, T, C, or G, that's the character state, right? So we're talking about comparing character states for shared characters across all the taxa that we want to compare. That's what the comparison is that we're doing. When you do phylogenetics, that comparison, you do a sequence alignment, and then you score the similarities and differences along a matrix, and then the phylogenetics program uses that matrix to make your trait. Basically, what you have to do is score ancestral and derived traits. So 2 more terms. I know there's a lot of vocabulary, folks, and I'm sorry. And Erica's sitting here like, I got to sit through this class again. I already did this
No,
in
no,
my life.
no. This is always good, always welcome.
So an ancestral trait is a trait that is old. It predates the group that you are looking at, your what's called your in-group, your study group. So for primates, since we're talking about primates tonight, an ancestral trait for primates would be having hair on your body, right? Because that's a trait for mammals, and primates are a subset of mammals. So being covered in fur or hair is an ancestral trait for primates, right?
A simpliciomorphy, if you will.
A plesiomorphy is the sciencey word you would use for that. You would use plesiomorphy instead of ancestral. And then a derived trait is something that is new in the clade that you care about, right? So for example, for it's not all primates. Erica, you know better than me. What's the clade of primates for opposable thumbs? What's the big word for that group?
Well, actually, most primates are going to have opposable thumbs in
Yeah,
the sense that they have they have the musculature that's needed to actually do that gripping. And for all primates, for instance, like a characteristic that would be unique to all primates as compared to mammals, as like a suite, as a synapomorphic characteristic for all mammals, it would be like 5 digits on, or not 5 digits on their hands, highly mobile ankles and wrists, right? Or binocular color vision, typically, or a reduced sense of smell, like lowered olfaction. There's lots of different things that you could point to for primates, but these exist each and every taxonomic rank as you go up or down. So as Dan's talking about, like assessing these ancestral versus derived characteristics is also context specific of like where you're looking.
That's exactly it. So ancestral and derived, the way I always say this in my classes, these are relative terms. So you look at one clade, it's going to have a set of ancestral and a set of derived traits. If you zoom in further, the derived traits for that, say, primates, right? Those traits are now ancestral for apes, for example, and then apes are going to have a new set of derived traits that set them apart from the non-ape primates, right? So as you zoom in and out along your phylogeny, what counts as ancestral and derived is different, right? So looking at this paper, what they're going to do is we're using our ancestral and derived traits. We have our taxa that we want to compare, and the test we're going to do is this permutation tail probability test. And here's the fun part, because the goal here is to generate the shortest possible trees, right? The most parsimonious trees, right? How do you explain your data that you're directly observing with the fewest possible evolutionary changes from your common ancestor? So in order to do this, you do this PTP test. And the way this works, and folks, this is where it gets complicated. This is where, even for me reading this paper, it twisted my brain into a pretzel and I had to untie that knot and go back and figure it out, right? So what they do is you randomize your data, where you are randomly assigning character states for each character to your taxa, right? You're randomly assigning it. And then for the randomly assigned data, you're building a phylogenetic tree that is the most parsimonious tree possible. And then you do it again, you randomize it again, and you build another most parsimonious tree, and you do it again and again and again. And the point here is to generate a distribution, right? You want to generate a distribution of tree lengths, because the question you're trying to answer is, what are all of the possible tree lengths I can get for this set of taxa with these set of characters and character states? What are all the possible ways I could arrange these data to get a certain tree, right? A tree that has whatever minimum length. So you randomly allocate your character states and you build a tree and you calculate its total length. You just add up the length of all the branches. And then you do it again, and you do it again, and you do it like 1,000 times. Now, in some of these older papers, they talk about doing it 100 times, which I thought was adorable. In the modern world, when you do this, you're doing it a minimum of 1,000 times. More likely, you're doing it 10,000 or more.
That's the baseline that I've always heard. Is do it 10,000 times. 10,000 times at least.
Yeah, and that's going to get you a distribution of basically all possible trees. And as they say in several of the, I think it's both of the old papers, they say, look, we're not necessarily going to sample from the entire distribution, but by randomly sampling from a lot of the distribution, we can accurately reconstruct what that distribution looks like. So at the end of this, you get statistics for possible tree lengths with the data you're dealing with. You get a minimum and a maximum and a median and a mean of like, what are just all the possible ways these data could be arranged, and what do those trees look like in terms of their parsimony. Some of them are going to be quite parsimonious. They'll be very short. Some of them are going to be a mess, and they're going to be very long, non-parsimonious trees, right? It's just how you randomly arrange your data. Now, the key thing here, oh, go ahead.
No, I was going to ask, just before we go too much further, what would be like a good, let's say you're explaining it to a 5-year-old, explain it like I'm 5. What would be a good analogy to use here for what's going on, like what we're actually doing?
Yeah. So the analogy that I like for this, it's not doing the same thing actually statistically, but conceptually it's doing the same thing. Is, Erica, I'm sure you've had to take at least one statistics class in your life. And chat, anyone watching this, if you've ever taken a statistics class, perhaps you've done this thing where the teacher, they have like a bag that has a whole bunch of marbles in it, right? And there's like a whole bunch of marbles in this bag. Everyone take 10 marbles, right? And everyone takes 10 marbles out of the 1,000 marbles in the bag. And then you go around the room and everyone tries to guess what the frequency of each color is, right? Red, green, blue, yellow, white in the bag based on the 10 that they picked, right? So you get a whole bunch of samples. And you don't know what the actual frequency of the marbles in the bag is. But everyone's got a little sample, and everyone can make a guess based on that sample. But then what you do, and this is the part where the teacher blows everyone's minds, is you pool everyone's data together. Instead of having 20 individual samples of 10, you do a single sample of 200 marbles, graph that, and then the teacher reveals what the actual distribution is, and it's going to be quite close to that larger sample, right? So the idea is we want to randomly sample from an unknown distribution in order to probabilistically determine what that distribution looks like. That's fundamentally what we're doing with this technique.
It's like running a bunch of tests on a microcosm of a ton of data, just like so much that it's computationally very difficult to assess. You run the stats on a microcosm of that data, and you're going to approximate, theoretically, the statistics, the general statistics, mean, median, et cetera, for your larger data set. That's the general idea.
Right. And then here's the actual test. So generating that distribution of all possible minimum length trees, like all possible most parsimonious trees with any configuration of your data, right? You generate that distribution, and then the key step is you're going to compare that to your actual most parsimonious tree with the real data as those sequences are allocated to the taxa in real life, right? So you're generating a set of branch lengths, essentially, and then you're saying, where does my real data fall in that distribution? If it falls inside that distribution, then that's basically saying you don't have a real phylogenetic signal in your data because it doesn't differ significantly from randomly allocating those character states, which is to say when you randomly allocate them, right, there's no phylogenetic signal. There's no shared inheritance pattern there, right? So you're disrupting the phylogeny, seeing what all the options are, and then seeing if your actual data have a strong phylogenetic signal in them, right? That's what we're trying to accomplish here. How do we feel? Like,
it's a lot. It's a lot. Listen, I just want to say, like, over the past 2 weeks when we've been digging into this, and Dan has done the overwhelming bulk of the work diving into this, and he'll message me and explain it, and I would be like, okay, okay, I need to read this about 12 or maybe 13 more times, and then I'll start to wrap my head around it. But once it clicks, it clicks, and then you're like, okay, I see what we're doing.
You know what? Here's what we're going to do. I'm gonna call an audible here on something that I was thinking about doing, and I hadn't decided to do it or not because we're done with Archie, so I can take this paper down here. So I'm going to call a little bit of an audible here. I'm going to pull up. I'm just going to draw a little bit, and I think that'll help us. So if everyone just wants to just give me a minute to just play around with my screen a little bit here, I'm going to take this down. Okay. And just give me a second. I'm going to do this on PowerPoint because I do for class, for simplicity's sake. Here's a little peek behind the curtain of my real job here. For simplicity's sake, I don't use any fancy software like Illustrator when I'm making figures for my classes, because I will modify my stuff on the fly right up until the moment I'm teaching the class. Like if you look at my PowerPoint slides for class, it'll be like my class starts at 3:50 in the afternoon. It'll be like last modified 3:43 p.m.
Yeah.
Like, so I do everything on PowerPoint, right? And just as just a rule of thumb, so that it's plug and play and I can just mess with it no matter what I need to do. So I'm going to share my screen here, and let's go to, no, I want to do this. There's so many things I have open here. Share screen. So
you're preaching to the choir. I've
so
got
now we're going to share this PowerPoint window. Okay, so now we're going to do here is I'm just going to go into the drawing here, and I'm just going to futz around, because what we're going to do here is we're going to draw. We're going to do this. I want it to be black. I don't care. So what we're going to do here is we're going to have one axis. This is going to be frequency, right? And then over here, this is going to be length, which is to say parsimony, right? So on the left is going to be more parsimonious, which is to say shorter trees, and on the right will be longer trees, which are less parsimonious. Now, what you're doing is you are randomly generating phylogenetic trees and calculating the most parsimonious configuration for each one, right? You're allocating the data randomly, generate the most parsimonious tree. Okay, well, it was this long. Okay, you do it again. It was this long. You do it again. It was this long. Then it was this long. Ooh, that was a good one. It was that long. Oh, that one was really bad, right? And you're going to start clustering, right? I'm treating this almost like a histogram, right? You're going to start clustering, and you're going to generate ultimately a distribution of tree lengths. And then what you want to do is take your actual tree, whatever those data look like, and say, okay, what's the branch lengths? What's the overall length for the most parsimonious tree? Does it fall here in the distribution? Does it fall here in the distribution? Or is it like a super outlier? Does it fall over here, right? And if your tree falls somewhere in like the meaty part of the distribution, that means it doesn't differ significantly from a random assortment of character states, which indicates a weak phylogenetic signal. Whereas if it is, oh, go ahead.
No, I was going to say, like, to dumb it down as much as I feel comfortable doing, right? You're basically saying, this is what our real phylogenetic tree looks like. Is it real or is it artificial? And in order to test whether it's real or artificial, you're like, what would a gazillion random phylogenetic trees look like? And if none of them can perform anywhere close to as well as your real phylogenetic tree, as far as parsimony goes, then you're like, okay, it's probably real, because randomness can't get the same thing.
It doesn't do, right.
Yeah, you could do the same thing like at the beach, where you take 1 million kids and blindfold them and have them try to draw something, and then see if it how it compares to an actual drawing that you made, right? To see if you could produce your drawing with random chance. That's kind of what you're doing here. Probably butchered that, but that's how I understand.
That was excellent. No, that was excellent. Right. If your real tree doesn't differ significantly from the random trees, then that tells you there's not much going on in your data, right? If your real tree differs significantly, right? And, we'll talk about what level of significance matters. You could argue about p-values and stuff and what statistical test you could use. At the differences we're going to see, it doesn't matter, right? This is going to be, as we'll see, the epitome of getting data that's so good you don't need to do statistics on it, right?
To be clear, the data is so good that when James Tour, the creationist who was originally putting his 2 cents in on all of this stuff, when he saw these p-values, the quote, this isn't like, I'm paraphrasing, but he was basically like, it's too good. You don't see data this good, right? Meaning there must be something wrong with it.
Yeah.
Because it so routes separate ancestry.
So this is our first pass on the PTP test, right? So this is just Archie, right? You build your distribution and then you evaluate if your phylogeny falls within or outside of that distribution statistically. That's our first pass on the PTP test. But we're going to go to that second paper, Faith and Cranston, because I think they do a really nice job explaining it slightly differently. And I think it's really clear. So let me take this down. Let me share my screen again and go to the Faith and Cranston paper from 1991. Here we are.
Give thumbs up in the chat if you felt that beautiful normal distribution that Dan drew helped you better understand this concept. Because I thought I'm a visual learner. So to me, I was like, oh, okay, it actually makes, it feels more intuitive when you see it than when you're
I was sketching things out for myself the other day. I was like, all right, I see where this is going, especially when we get back to bomb, because for the tests of common ancestry, it's one thing, but then for the separate ancestry, it's a different thing. We'll get there, and it's useful. Okay. So now we're going to look at this paper from 1991 by Faith and Cranston. Could a cladogram this short have arisen by chance alone on permutation tests for cladistic structure? And I am a professional, so I'm going to remember to add that to the screen so everybody can see what I just read. I meant to have that on as I was reading it. Here we go. So this line just like really helps, I think. The cladistic permutation tail probability, PTP, is defined as the estimate of the proportion of times, I'll zoom in a little bit, that a tree can be found as short or shorter than the original tree. And by original tree, we mean the most parsimonious tree you can make with the real data. How frequently can we find a better tree using randomized data? And if it's a lot, then your real tree is no good, basically. That's what it's telling you, right? So another way they describe this, they go down here. They're a little more technical here. They describe parsimony and why parsimony is useful. And I also, for everyone watching, found that actually very helpful. So if you want a good explanation of why parsimony is a reasonable criterion, this is a good paper for that. So we're going to go down here, the cladistic permutation tail probability. And I want to highlight this line. The observed tree length is compared to a standard defined by the theoretical minimum number of steps possible with the data, right? You randomize your data, so you're getting every possible configuration of those same data, right? You're preserving the frequency of ancestral and derived traits, but you're randomizing the allocation. What do the best possible trees look like? And I want you all to put a pin in that line, right? Because we're not just comparing random trees, we're comparing the best possible trees to our actual distribution, right? That's a key point that we're going to come back to later. So everyone just remember that line right here. Okay.
Write that down. Write that down.
Write that down. It's on page, whatever page this is. I don't even know what page this is. I don't even know if it has
exam.
pages in it. Whatever. So here is, let's see, I want to find a specific line here. Where is it? I'm trying to find the line that reads, I'll read it to you because it's in my notes, but I'm trying to find it in the paper. They define the, I'm sorry?
These beautiful block quotes.
I know. I highlighted so much. Oh, here it is. PTP, this is the line I wanted to read to you. The proportion of all data sets, actual plus randomized, producing a tree as short or shorter than the length of the actual tree. In other words, we're comparing our actual most parsimonious tree made with real data to the distribution of all possible trees from the permuted data and saying how many of those are shorter, which is to say better than the real tree, right? That's the test we're doing.
This is the number, like the quantitative evaluation of the demonstration that Dan just gave, right? So it's basically, this is the number that test is going to spit out that you can use to empirically assess you, like that's what this test is doing.
Yeah. So a paraphrase of this is to say, if we randomize the data and we randomly allocate those essentially nucleotides, right? We're randomly allocating nucleotides for each shared location in the genome. How often from those randomized data can we make a tree that's more parsimonious than the tree we can make with the real data? And again, the point here is to evaluate the robustness of the phylogenetic signal in the real tree. Because if your randomized data are doing better in terms of parsimony than the real thing, that's indicative that the real thing is not actually showing you a strong evolutionary relationship, right? But if the real thing is way more parsimony, if it's this big outlier compared to your randomized data, then that's indicative that that's an actual evolutionary history that you're looking at. and not just an artifact of like convergence or something.
Tell me if this is a useless analogy, but maybe this will put it in a context that is maybe a little bit more understandable. Like, let's say I have a group of 10 people and I want to assess if those 10 people are actually related. I want to make a phylogenetic tree of them. They're of a bunch of different ages. I want to see if they can form a family tree. So I sequence all of their genomes and I run that through and get this nice phylogenetic tree using the entire genomes of all of these people. Then using this test and these random character states, right, to generate all of this random data, I generate all of these kind of noisy, useless phylogenetic trees that should not be indicative of anything real because it's noise, because it's random. We wouldn't expect by chance to get the same signal that we would from a real phylogenetic tree made by real people showing a real relationship. So then when I compare those two, if I look at my distribution and I find a whole lot of trees that look like the tree that I generated from these 10 real people, then they're probably strangers. They're probably not actually related to one another. It's probably noise. But if the tree that I have for my 10 people looks really nice and is very short and very parsimonious compared to all my noisy generated trees, then I can probably be confident that I'm finding a real phylogenetic tree. These people are all really actually related because it performs better than randomly generated noise.
Yes, I think that's perfect. I think if you were to take like 10 people who are, say siblings or cousins, right? It's a mixture of siblings and cousins, right? It's 2 siblings and then 4 siblings and then 3 siblings, right? Yes. You take an actual family of siblings and cousins all in the same generation versus 10 people who are, say, outside of third cousin from each other, right? You would be able to do this test and tell which set of people is which. Yes, I think you are exactly right that is a thing you could theoretically do here. Because, as you said, the actual family should have a very tight, very parsimonious, very consistent phylogeny that you build. Whereas the unrelated individuals, you could permute that data and you would get trees that are equivalently parsimonious compared to the real data because they actually don't share close relations.
Right.
Another way to think of this is you could build that distribution and then you would plot those 2 trees, right? And one of them would fall in the middle of the distribution and the other one would be a really big outlier on the short end.
It's going to be, exactly, because that's going to be your
cool,
well-performing tree.
Okay, that was awesome. Thank you for that analogy. Chad agrees. That was awesome. That helped.
Good.
So now we're going to go back. We're going to take this down. We're going to go back to the recent paper. We're going to go back to Baum.
Now we all understand PTPs. We're all on the same page. We all know exactly what's going on, including Rob, who is watching from afar. Rob, I hope this is helpful.
All right, so now we're going to go back to Baum because Baum is going to add some wrinkles here that we haven't talked about yet, right? They're going to apply that test in a unique way, right? They're going to do something a little bit different from what we just talked about, but that's the test that they're using to evaluate the data that we're dealing with here, okay? So the quote from Baum that I want to highlight here, and let's see here. It is, I'm just going to Control F this so I can find it and have it on screen when I read it. Evaluate, there it is. It's right in front of my face. This is the line right here we want to use to evaluate whether the observed hierarchical signal is more than expected under species or family separate ancestry compared to common ancestry. So this is where this gets useful in terms of the thing we want to evaluate, the robustness of common ancestry, right? We want to evaluate if we hypothesize each species that we're dealing with has a separate origin, or each family that we're dealing with has a separate origin. We're going to use this PTP test, and we're going to compare that to a PTP test using the hypothesis of common ancestry. So we're going to do this, we're going to do separate tests here, and we're going to compare the actual phylogenetic tree to the expected distribution, okay, in these separate cases. And it's important to note that we need to use separate distributions for each test because they are fundamentally different trees, right? With the family separate ancestry, you have a bunch of independent families, right, that have common ancestry within the family, but no actual branches connecting them together. With common ancestry, you have one tree for everybody. And then with species separate ancestry, you're looking at each individual branch, right, on its own. So we're not the more technical way to say this is each of the 3 tests is sampling from a different distribution, right? It's not, you can't sample for the common ancestry phylogeny from the family separate ancestry distribution because they're actually separate data that you're working with.
And I think it's worth noting, and I don't think that this is going to be over anybody's head. I think we're probably all on the same page here, creationists included in the audience who may be watching. These 3 hypotheses, these 3 models produce 3 specific different like predictions, right? Like we should see 3 different things that are distinguishable from one another. That's just the way that it is, which is nice. But we'll get there.
So here is one more wrinkle we have to add here, folks, and it's going to lead to another wrinkle because this is how, by the way, this is like a good peek into like what graduate school is like, where you're just reading papers and then you go, oh my goodness, what is going on? I have to go to this other paper to figure out what this method is. And then, oh, I got to go to this other paper. So this is what we're doing here, folks. This is Erica, you know, you've done this more recently than me, but like, this is what we do in a graduate seminar. Like this is journal club, you know?
This is journal club. This is every symposium, every seminar class that I've ever been to. I had to do this recently because I've been trying to learn to the best of my ability what I'm doing in my dissertation, because when you run into little roadblocks and when your methods are new, or at least they're applied to something new, there's nothing to refer to. Like you have to figure it out yourself. And so you have to do it. Papers that are similar, but they're not identical. So you have to be like, okay, well, how does this apply to what I'm doing? And it's a game of trial and error. And then before you know it, you're reading a paper from 1985 that is doing the same thing, but by hand. And you're trying to figure out how this maps onto what you're doing. It's a great time. Go to grad school, everybody.
It's awesome. It's so cool. So now we get to this next part, the second half of that sentence, right? To evaluate whether the observed hierarchical signal is more than expected under species or family separate ancestry. That's SA is separate ancestry. We use the PTP test, which uses a Monte Carlo approach to simulate data under the SA hypothesis. So what in the world is a Monte Carlo approach? Now we have to figure out what that is. Okay, so a Monte Carlo approach is a method for determining a distribution for a set of data. In this case, the minimum possible tree length of all possible trees given our randomized data. I basically showed a Monte Carlo approach a few minutes ago when I kind of started doing that little histogram with the dots and then just drew the curve. That's a Monte Carlo approach, right? You are sampling from some unknown distribution in order to generate that distribution. And you're never going to perfectly do it, but the more you sample, the closer you can approximate that distribution. And there were some really cool real world applications of this. Like one of my favorite examples of this, and I love this example, and if you go to the Wikipedia page, it's right on there for Monte Carlo methods. You can converge probabilistically on the value of pi using a Monte Carlo method based on the fraction of points that fall inside and outside a circle. It's really cool.
Oh, math.
Yeah. Math. You could just randomly drop points on a plane with a circle, and basically the frequency of inside versus outside, you just do, and it converges on the value of pi. It's so cool. It's great.
So Monte Carlo is quite Bayesian, yes?
This is a Bayesian approach.
Yeah,
yes,
okay.
Exactly. So the whole point is we're going to sample big numbers to converge on what a correct value is. And this is the part where creationists are going to quote mine me. Ready, Erica? You ready for it? This is the part you're going to quote mine, everybody. We're going to converge on a correct value that is, in an absolute sense, unknowable. But we can statistically, probabilistically converge on that value with a high degree of confidence by random sampling from the set that build that distribution, right? So does everyone see that how creationists are gonna cherry pick that one line? They go, oh, they can't know the answer when they do phylogenetics, right? There you go, folks.
Yep.
You just get the sentence on either side; they'll get it. All right. So we can't know the actual distribution, right? There exists this universe of phylogenetic trees with the data we have that all have minimum lengths. We don't know what that universe looks like, but we can simulate it by randomly generating those trees or randomly sampling from the distribution. Those two phrases mean the same thing. Go ahead.
And to be, yeah, no, all I was going to say is that it's like the reason that this is unknowable, right? In essence, is because we're dealing with such enormous numbers here. Like, I
Yeah.
mean, we're talking about in a molecular sense, we're talking about the number of mutations within the order primates. Like, it's just obscene how many possible trees there are for this, right? But you can converge on a really nice approximation. I mean, it's like Dan was just saying with the example with pi, right? Like, you can approximate pi. You're not getting pi exactly, but you are getting close enough that you could use that in engineering, for
Right.
example.
Yeah. You can
Anyway.
you can find online, like, use Monte Carlo to simulate pi. If you Google that, you'll find simulations, and you just watch it go for a couple minutes, and you'll have like 5 decimal places.
Yeah.
Right? It's really cool stuff, folks.
It's close enough. True, in in a real sense, it is close enough.
Yeah. So I'm not going to go through this in more detail because we kind of talked about it with the marble thing with the bags. I drew out the curve and showed that's what a Monte Carlo simulation is. But there is, Erica, one more wrinkle we have to add, because when you do phylogenetics, Monte Carlo is just one MC that you're going to use. There is a second MC that is also going to play a role here. Yeah, because these are fundamentally MCMC methods. They are Markov chain Monte Carlo methods. And Erica, if you want to do Markov chains, I will let you have it, because you seem to really like Markov chains.
No, I mostly,
and I'm happy to do it too, either way.
I just have a fun analogy that I think helps you understand Markov chains. So you give the basic explanation, because you're the guy who knows this better than I am, and I'm probably going to use a wrong term, and then Stadler's going to get me on it. So I'm going to let you handle that, and then I'm going to give you the analogy that I was taught when I took my first stats class about this.
Yeah, so a Markov chain, and I'll just preface this by saying, if anyone's familiar with the concept of a drunkard's walk, that's a Markov chain. If you know what that is, that's a Markov chain. It basically means it's a series of events where each possible next event and the probability of each possible next event is entirely determined by the current state. Wherever you are right now determines what can happen next and at what frequency. So the drunkard's walk analogy is, you've got a drunk dude stumbling drunk between 2 bars on either side of him, equidistant, and he's going to randomly stumble to his right or left. And the game is to try to figure out which bar he's going to stumble into first, right? So if he's directly in the middle, it's 50-50. And he's 10 steps, right? It's 10 steps total. If he falls 1 step to the left, now he's 60-40, right? Now 60% chance going one way, 40% chance going the other way. Now from that 60-40 position, he can either go 70-30 or back to 50-50, but he can't jump to 80-20 the other way, right? And this is important for phylogenetics, because what you're modeling is a series of changes in DNA, right? You have to get from your ancestral state, whatever that is, to your existing genomes that you've directly sequenced. So you can't do big jumps, right? You have to do one mutation at a time to get to your descendants. So when you're doing phylogenetics, they reference in this paper the Markovian, the Markovian power, whatever phrase they use, something Markovian. Basically, it just means it's a stepwise process of getting from your ancestor to your descendants. So when we describe these techniques as a Markov chain Monte Carlo, what we're doing is we are sampling from possible series of steps. Right? This is a possible series of steps. This is a possible series of steps. That's a possible series of steps. And trying to find, in this case, because parsimony is our standard, the shortest possible series of steps. Right? So that's what we mean when we say these are Markov chain Monte Carlo simulations.
So that's way more fun than my example. I wish I'd been taught that example. Mine was that you're trying to get to the top of a mountain, but you can only look down directly at your feet. So you can
that's good
vicinity. And so it's like, you pick the highest point that is within your frame of reference, and you go to that highest point. And then from that next point, you pick the highest frame of reference and you go to that point, right? But there's like some variability there. And so the idea is that each step, each pathway that you're going to take up the mountain is going to be like dependent on your starting point and each subsequent step along the way. But I like yours better. It's kind of like, that's very Bayesian as well, because it's like it's basically this idea that every step that you take is informed by previous information.
Yeah.
Right. So it's like, if I were to give you 2 jars and or rather, let's say I give you one jar and I tell you that this jar is I don't know, 90% red marbles and 10% green marbles. But or it might be the inverted, right? It
It's
might be
one or the
90%
other.
green marbles and 10% red marbles. And I say, which do you think it is? And you're going to say 50-50, right? 50-50
Right. Who
chance.
knows?
But if I pull out a red marble and give it to you, all of a sudden you're going to be like, oh, no, it's probably much more likely that we're
Right.
looking at
But then,
a jar of marbles that's mostly red.
right. And then each iterative marble you pull becomes more informative, right? I put red, red, ooh, green, red, green, green. Ooh, now we're drifting back towards 50-50, right? Each marble becomes more informative. And again, I hope everybody can see the relationship to what we just talked about of converging on the correct distribution, right? It's thematically the same thing where as you go, you become more confident in your conclusion, right? You get closer to one door or the other, right? So that's what we're doing here, folks. We're using a Markov chain Monte Carlo simulation to probabilistically determine all of the possible shortest phylogenetic trees for a given randomized data set. And then we're going to compare that distribution to the actual most parsimonious tree for the real data.
I am so pleased at the amount of people in the comments who are like, yeah, that makes sense. that's the best thing you can possibly hear when you're trying to explain a topic that is difficult and you're honestly not even sure if you're getting it 100% correct. So that's amazing to hear.
I have spent so much time this week practicing how to explain this in a way that makes sense to people who have never heard any of these words before. Yep. I'm trying, I just, if it's not getting across clearly, I apologize. I just want you all to know I'm trying so hard.
I was puzzling over it this afternoon too, because I was like, because we were discussing how we were going to present this to you guys, and I was like, okay, let me think, because for me, I thrive off of analogy, so I was like, okay, well, how can we actually pitch this in a way that is intuitive? And then Dan had the idea of the marbles, and so it's like, okay, let's keep things marble and drinking centric. Then it
Look, I didn't have that idea. I did that in my 12th grade statistics class. I remember doing that, taking the marbles and trying to guess, and then we got the right thing if we pull them all together.
My stats class. Yeah. My stats class was like, we're learning R today, and that's how you're going to learn stats is by trial and error in R.
That's more useful. Let's be clear. That's more useful.
It's helpful once you get there, but those first couple of days, I was like, I don't think I'm cut out for this.
I wish I had done that in 11th grade, but we digress. Okay, so let's just kind of recenter ourselves at where we're at, right? We are using a Markov chain Monte Carlo or MC method to find the distribution of the most parsimonious trees for randomized data, comparing that distribution to our real data, the most parsimonious tree or trees, because for separate ancestry, that necessitates many trees, right? Tree or trees for our real data. And then we want to know if those are members of the same distribution or if they are different from each other, right? So now we can get into what we're actually doing here, okay? So now what we're doing, I'm looking at my notes going, okay, how do we want to approach this? What do I want to be showing on screen? I think what I'm going to do is I'm going to go back to the PowerPoint slide, actually, and we can draw this out. And then I'll come to the data tables for the paper. So we'll draw it and we'll do the data tables. Because what we can talk about is we could do, okay, here's what they did for common ancestry. Here's what they did for family separate ancestry. And we don't really have to worry about species because nobody thinks that except for one rando on YouTube.
Literally,
if you know, you know.
You know who we're talking.
Right.
Dude,
other than that,
did not descend from wolves. They did not.
Yeah. Other than that, we can just ignore, nobody thinks species separate ancestry is a thing. So we can just basically ignore that. So what I'm going to do is I'm going to take this down and I'm going to reserve the right to bring it back at a moment's notice if I get confused. But now I'm going to share the screen. We're going to go back to the PowerPoint window, which I have to, wait a minute. I have to find it. There it is. Okay. Now I'm going to share my screen, window, PowerPoint window. There it is. Now I'm going to erase everything here. We're just going to erase all this. Oh, that's going to be a pain to erase. Sorry, everybody. Not well planned. Okay. because we're going to do this fresh. We need to have this fresh, and we're just going to draw distributions. We're not going to worry about deriving the distributions. Now we care about comparing I should leave the axis. We're going to worry about comparing the distributions to actual data. And I'm going to make this a nice long x-axis.
Wait, Dan, are you hoping you're sharing this? Because you're not sharing it.
I'm a professional. Thanks, Erica.
Okay.
Thanks, everybody.
At first, I wasn't sure, and then I was like, "Oh, he definitely."
Yeah. What I did is I hit share screen, but then I didn't add it to the screen when I did it. All right.
So this time I've been there.
So this is going to be length again. Okay, and again, length is going to get bigger. And so now what I'm going to do is I am going to do. We're going to do common ancestry here. Okay. Oh, I can have multiple colors. This is great. Okay, so we're going to make this one blue. Make this one a nice blue. There we go. Okay, so we're going to do our common ancestry. We're going to say that's going to be too light. We're going to do this in like I can't use green. We'll do purple. Okay, so this is going to be common ancestry is going to be in purple. Okay, everybody, that's CA. Common ancestry is going to be purple. So we're generating a distribution here, right? Like this. I'm calling it approximately normal. I forget if it's actually an approximately normal distribution, whatever. I always draw my normal distributions weird on the right side. Don't worry about it.
Beautiful.
It doesn't matter. We're going to call this this is our randomized data with common ancestry. So we're saying all of these taxa share a common ancestor. We're going to randomize the data, and we're going to plot every most parsimonious tree. I forget how many it is. It doesn't matter. Just we're going to plot a bunch of the parsimonious trees, right? That's what we generate our distribution, right? They have, and if you go to, there's a figure in the paper where it has the minimum, the maximum, the median. It has those three parameters, and then I think elsewhere they talk about the mean and the variance for each one. But in the table that I'll show you in a minute, it has the minimum, the maximum, and the median for each distribution. And importantly, it has the length of the actual tree. Now remember, for common ancestry, what we're looking for here is a tree that is significantly shorter than the randomized data, right? Because that is indicative of an actual phylogenetic signal. So
it should plot way off to the side, away from this distribution.
Right. It should be not part of this distribution. And that is, in fact, exactly what they found, right? So if you were to plot this, you have your parameters of whatever your tree is. You get your minimum, right? Your maximum. You've got your mean or median right here. The actual data was like all the way over here. It was a giant outlier. It wasn't even close, right? Huge outlier. Now, so that is strong evidence for common ancestry, because what this means in our test is the randomized data, right? The distribution that we have, there's no actual phylogenetic signal there, right? And if we just randomly shuffle the deck and deal the cards, right? What are the best hands we can get? Or if I take the deck out of the package new and I go to the end of one of the suits, what's the best hand I can deal, right? It seems like something weird is going on there. I always get royal flushes when I do that. That's convenient, isn't it, right? So here, right, you have your real data that is a significantly shorter tree, right? The branch lengths in total, when you sum them up, are way shorter, which is to say there are fewer evolutionary changes required than any possible configuration of your randomized data, which is to say there is a strong phylogenetic signal of common ancestry in the actual data, right? That's what that means. We've done a PTP test and established that common ancestry is strongly supported. So that's good. Let's
shoot off the fireworks. We're happy. We're good. This is a big classic common W for conventional science. About to send creationism into early retirement. But we still have to see what we get for separate ancestry, right?
We have to test other stuff. So what
if they both do just as well?
Let's change the game. Let's test family separate ancestry. Sorry, I'm drawing with my mouse, so I can't draw letters with my mouse. I apologize, folks. It's bad. Anyway, FSA is family separate ancestry. Now, here's the thing that we're looking for here now, right? The key thing with family separate ancestry is there is, in actuality, according to this hypothesis, no phylogenetic signal between the families. So what they did here is it's a little bit tricky explaining what they did for family separate ancestry. So here's what they did. Generating the theoretical distribution works the same way, except they're positing independent ancestors for each family rather than one single ancestor. So when we look at the tables in a few minutes, the FSA trees, all the numbers that we get are smaller because, again, we're adding up branch lengths and you're missing all the internal branches, right, that connect the trees. So you just have more branches in the common ancestry tree versus the family separate ancestry tree, right? So the overall numbers are going to be smaller. So the x-axis here with the two colors is different. It's not the same numbers. I'm going to overlay them so you can see the patterns. I'm going to leave the purple on there, but it is a different distribution and they don't actually overlap with each other, okay? And that's important to know.
Why, by the way, they couldn't use the same distribution for both.
Right.
Like because you're fundamentally testing two different things.
You literally can't get the same branch lengths on the separate ancestry tree because there are branches, depending on how you want to conceptualize it. There's two ways they do it in the paper. You can either say there are branches that don't exist, right? Because they're separately created. Or another way to think of it is, right, there's no phylogenetic signal across the different groups because they were separately created. Another way to conceptually describe that phylogenetically is that they are connected by branches of infinite lengths. And I'm going to say that, and I'm just going to leave it there, and we're not going to worry about it, because if you think about it, it might twist your brain into a pretzel again, and we don't want to do that. So don't worry about it. It's a way to conceptualize it. If you're down with phylogenetics, I promise you it makes sense, and if not, don't worry about it. So I'm going to draw another distribution, because what we're showing here is a distribution. Now, this one's a little different. This distribution is based on randomly sampling one species from each family and then doing the data permutation, right? So there's several levels of randomization here where you are sampling one member of each family and then you're doing the randomization that we talked about. The reason you're doing that is because within each family, you have an actual phylogenetic tree.
And that everyone accepts.
That everyone accepts.
We're all okay with the trees at or below the family level. Creationists, conventional science, we're all good with that, except for one person. But the branches that join all of those families together are real in common ancestry, and they are an illusion in
exactly. That's a great word. And that's something we're going to talk about in a little bit. Basically, the illusion of common ancestry where it doesn't actually exist according to the separate ancestry model. So the key thing here is we're generating a random distribution of many trees, because again, each family gets its own tree, right? Many trees, and we're connecting them. And what we're trying to do is generate the most parsimonious set of trees. Now, here's an important thing for this, right? The data that we're working with are the extant taxa. We are not characterizing anything above that. This is critical to understanding how these methods work, right? We are generating for every data set, real and randomized, what the most parsimonious tree is for that individual data set. That means that each common ancestor of each family, and I'll draw it here, what we're doing here for family separate ancestry is we're going to have like a tree, and then we're going to have another tree, and then we're going to have a third tree, and then another one. And the point is they don't actually connect to each other. Whereas for common ancestry, right, it's one tree. And you're just going to keep, connecting everything in your one tree all the way down to the individual species that you're dealing with, right? It's one tree as opposed to many unconnected trees. Now, the configuration of those roots, those ancestors for each tree, we're not saying what those look like, right? We're randomly, or based on our data, right? We're sequencing these and then randomly permuting them, we're changing them, and we're letting the data tell us what the optimal configuration is for each simulation,
So you can kind of think of it brutally like you've got your observational present day sequences, or if we were including them, biogeographic data points,
Right.
or fossils, or morphologic features of every species. You're taking those and working backward, in a sense. In a sense,
Yes.
that's not literally what you're doing, but you're generating all sorts of possible trees that could potentially give you what we see now. So there is no there's no taking a state, making presumptions about it, and evolving it forward,
Right.
right? And
Right.
critically, again, this works in areas that everyone agrees, right? This method works within human populations, or works within dog breeds, or works within Felidae, whatever. That's the linchpin of all of this, in essence.
Exactly. And we got a couple really good questions in the chat, actually, that I'm going to pull up that can help clarify this here. So one here, that's not in the right place. Hang on. Here we go. How do we compare the parsimony of a set of FSA trees to the parsimony of a common ancestry tree? Do we sum the branches, average them, something like that? They are independent tests. We are testing each hypothesis independent of the others, right? So we can say common ancestry is well supported by the data. Then we can go and test family separate ancestry and say, is that well supported or not, right? These are separate tests. I should leave that up for a second while I'm saying it. These are separate tests.
The way that you're comparing them is by comparing their performance independent of one another, right? So you're not shoving them both in and running a stats test on all three at the same time. You're saying, given the data, how well does common ancestry explain it? How well does FSA explain it? How well does species separate ancestry explain it? And so on and so forth.
And the reason we have to do that goes back to the idea that because of the different hypotheses, that necessitates different distributions, right? It is just simply a different distribution from a series of trees with separate ancestry compared to a distribution of trees with common ancestry. Those are different data sets, right? So it would be inappropriate to compare the common ancestry tree to the distribution of family separate ancestry trees. That wouldn't be a meaningful comparison. Right?
You could think of it like this, right? Like, let's say you were trying to test the efficiency of the metabolism of two different organisms that have metabolisms that run in different ways. Like that, let's say you're comparing like an arthropod to like a mammal or something like that. I don't know very much about metabolics, but I would guess that the way that you would assess like a tiny little arthropod versus a big, huge elephant or something like that are going to be different. So you can test their efficiencies within their metabolisms and get like comparable values, but you're not going to put them in a box and test them simultaneously because they're just different animals.
Yeah, different things. Yeah,
if that makes sense.
And then I just want to show this question right here. Is this still a Markovian process? Yes, you're just limiting it to within each family instead of all the way back. But the Markovian assumption within each family still holds of you're starting with a common ancestor, and then you have to stepwise generate your extant sequences within that family. Yes. So the same logic holds, but we're hitting a stopping point, right? Now, the fact that we're hitting a stopping point has implications for what the results mean, because what we're testing here is there actually evidence of separate ancestry, which is to say, is there a lack of a phylogenetic signal? So an affirmative result for the family separate ancestry test would be for the actual most parsimonious tree to fall within the randomized distribution. And this, we're going to pause on this for a minute because it's, again, not intuitive given what we just did, but it's related to the idea that we're testing separate hypotheses here. So in the family separate ancestry test, there is, in actuality, right, there's the hypothesis is there is no actual connection between these individual clades, right? So essentially what we're testing for here is, or let me put it this way, the question we're asking is, does the real data contain a stronger phylogenetic signal than you would expect if they were actually separate at the family level, right? That is the question we're trying to answer with the family separate ancestry test. So we're going to do the same thing. We're going to draw a distribution. Again, you could ignore the purple for this. I'm drawing it on the same axes for simplicity's sake, but the numbers would not match. It's just for simplicity's sake for graphics, we're doing it on the same axes. Okay, so you're going to draw that red distribution, whatever it is, right? Let's call it that. And then you're going to get your distribution for your actual data. Now, for this one, for the actual data, you don't get a single most parsimonious tree because there isn't one tree. It's a set of trees. So for the real data, again, it's what?
It's a forest, if you will.
It's a forest, if you will, right? So for this one, you actually get a distribution. So this is another reason you have to do these separately. It's actually two separate statistical tests. You're comparing a single outcome to a distribution versus two distributions. And the question we're basically asking here is, do these two distributions overlap, right? So here is going to be our randomized distribution. And what we expect, if family separate ancestry is well supported by the data, that the real distribution of most parsimonious trees will fall somewhere within or overlap with the randomized data. Because again, randomization obscures phylogenetic relationships. So we expect those two things to be similar to each other if family separate ancestry is correct. What we find is that they are extremely different from each other, right? Our family separate ancestry distribution is like over there. It's not even close. And again, I'll go back to the paper and show you the numbers for this in a minute. We'll go through it. But it's not even close, which means, again, in terms of what the PTP test, the permutation tail probability test, is telling us, it's telling you that there is a much stronger phylogenetic signal in these data than you would expect if they were actually evolutionarily independent from each other. That's what the non-overlap indicates.
This is my like super dumbed down understanding of it, right? Is that it's family separate ancestry, trees within families, noise outside of families. Under common ancestry's model, right? It is trees all the way down, all the way up, all the way down. So we would expect that under the family separate ancestry idea for this particular test, given the distribution of the families, right, we would expect to see noise, right? Like it should be showing us noise. It should fall within that distribution. But instead, it mirrors what we see for common ancestry because the signal is there. The trees are joined together. They're not separately created. They are distinguishable from noise because it's an actual real pattern. It's not a collection of strangers. It's a family tree.
I was just going to bring it back to that. That was perfect. Yeah, what we're doing essentially with this FSA test is we're saying we think this is 10 unrelated people. And what the fact that the curves are non-overlapping tells us is, oh, these 10 people are all actually cousins, is what it's telling us. Yeah, I think that's perfect. So let's
tie up our little bow.
So having walked through that, now let's bring it back to the paper itself and look at the tables where they show the relevant data, right? And
because, guys, like, I'm sorry to interrupt. I just, like, you have no idea how extreme it is. Like, on this little graph, it's like, oh, yeah, like, it favors common ancestry. Favor is too light of a word.
That is not, yeah. So I'm gonna remove this. I'm going to go back now to BOM, and my goal for this screen share is to remember to put the paper on screen after I have enabled the share. Success. So now let's do the big reveal here, everybody. Let's go down to the data tables. Well, first we could just read the results, because the results section for everything we just talked about, all the methods, everything we just talked about is this little bit right here. It's this. This is how they summarize the results, and that's it. As summarized in tables 1 and 2, PTP tests for both morphological and molecular datasets consistently disfavored both species and family separate ancestry. In all cases, the observed tree length metric fell well outside the range of values expected under the SA model. In other words, for either species or family separate ancestry, the actual most parsimonious trees you can make always fell well outside the range of all possible trees if you assume they have separate ancestry. That's what those sentences mean. And that's it. They didn't elaborate further. They were just like, we're going to trust the audience or the readers to know what that means, and we don't have to spend more time on it. Turns out that is not always a correct assumption.
And this is meant to be like a snappy, this is a cool result that dunks on family separate ancestry and dunks on
It's a good paper.
creationism, right? And in that sense, it's a good paper. But like, I've been, and yeah, I was going to mention this later, but like I've been corresponding with Dr. Stadler on this. We've been going back and forth, talking a little bit about this paper, talking a little bit about creationism and evolution. And I don't think that's a secret. I don't think he would be upset for me to say that I'm showing emails or anything. But it's like, I had to explain to him because he was basically like, why didn't you guys go through all of the steps of all of the methods right off the bat? And it's like, well, in the first video, it was a throwaway, this is a cool paper that dunks on creationism, right? In the second video, it was like, no, the straw man that you're saying this paper is doing is not actually the case here. But at the same time, I am not a phylogeneticist, right? Like I'm not an evolutionary biologist in the same sense that Dan is or that Baum is. So it's like, when I read a paper, in a perfect world, I would read the paper top to bottom and investigate every single source in its bibliography to make sure I understand exactly what they did and how they did it. But in fields that are not my own, when I've got a lot of other stuff going on, it's like I don't necessarily have the time unless it calls for it, like it did today, because I will always get petty when I absolutely have to. But it's like I'm not going to spend hours and hours and hours like breaking down the statistics of somebody's paper unless it's in my field and necessary for what I'm doing, or the implications are huge. Again, if I had all the time in the world, I would do that. But it's like now that we have done this, and I hope the methods are at least relatively clear to you, right? We're going to go over to what Stadler said here in a minute, like his understanding of this paper. But it's like, it's not as they're presenting it is.
It's not. Yeah, it's not what they're presenting it as.
To be fair, we could have pitched it perfectly like the first time, but honestly, I didn't think that it was going to come. I didn't think it was going to come to this.
Didn't think this was going to lead to a 2-hour video, but here we are.
Here we are.
Here we are. So let's look at these results. Now, the thing I want to point out to everybody is the only line on these two tables we care about is the first one, the Perelman data set. That's the molecular data set, the genome comparisons. All the other ones, you've got morphology. Some of them involve fossils, some don't. We're not going to worry about it. So what I want you to see here for this is species separate ancestry right here, and then family separate ancestry right here. And we're going to ignore species. We don't really care. But basically, it's a very large z-score, let's say. Very large z-score. Exceptionally large. We're not going to worry about that because whatever. But this is the interesting one, family separate ancestry. Now remember, with family separate ancestry, because of the way this works, you get a range of best trees based on the real data rather than one. So both the real data and the randomized data are expressed as ranges rather than a number compared to a range, okay? So it's a different statistical test. We don't have to worry about that. Point is, here is the real data, right? Our range is 11,170 to 12,707 with a median of 11,965. And the randomized data ranges from 16,619.6 to 19,775.2 with a median of 18,331.4. You will all note that these are non-overlapping data sets. They are non-overlapping. That is like the key takeaway.
It's not close.
It's not close, folks.
I've got GM on the brain, but it's like in space, these two things are really far apart, which, as we demonstrated, is not what you want if you're trying to support family separate ancestry here.
Exactly. So that is the takeaway from this paper, right? That is how they arrived at those data. We went through the methods, and it's taken us approximately an hour and a half to go through step by step and then go to the supplementals, or not the supplementals, the references, and kind of construct what they did. And I hope everyone has an idea now of how we made these comparisons, right? This is the key. So we can just show one more data table down here. And now we have the p-values of species and family separate ancestry. And you'll notice that some of these are 0, because I'll zoom in and show you what this says down here. P-value less than 10 to the negative 300 is denoted as 0. And you will see, as we go up here, we're looking PTP on the molecular data set. That's the only line we care about. It is 0. It is 0. So yeah, those distributions are non-overlapping.
So I Googled this just because I wanted you to have the comparison here, you, the viewer. So the number of atoms that are in the universe right now is estimated to be about 10 raised 78, right? We are talking about like unreasonably low p-values here.
It is unambiguous.
Negative 300. Like
the phylogenetic signal in the data is completely unambiguous. There is no way to generate the pattern that we observe with any configuration of separate ancestors. That is the key. There is no way to get a level of parsimony that is anywhere near what we actually expect, what we actually observe.
And we're going to talk about this in a second, but this is not a straw man of any scientific model in creationism. That scientific is an important word here, as we're going to see shortly.
Yeah. So this is where, so put a pin in that, everybody. We've completed part 1, our deep dive into Baum 2016. I'm going to go back up to the title so everyone can see it. Statistical Evidence for Common Ancestry Application to Primates. We've completed our deep dive into this. Now, the reason we did this is because Rob Stadler decided to go off on myself and Erica's collective understanding and representation of this paper. So he released this 37-minute video on Dr. James Torr's channel on, was it Sunday? I think it was on Sunday. Saturday?
Yeah, it was like a week ago. No,
yeah,
it was less than a week ago.
No, it was recent. Yeah, it was, I think it was over the weekend sometime. I think it was on Sunday as we record this. So it was a couple days ago. So now we're going to look at his video. Erica, are you ready?
I'm so ready, because I want you guys to know that when I listen to creationists who are covering me or one of my friends, right? I usually listen to them at the gym because the
science,
the frustration powers me to get my PBs, to get my personal best. But this one was like, and I'm sorry, Dr. Stadler, if you're listening, but like this one, it didn't even really make me that mad, because it's hard to be upset when you feel like you're grappling with somebody who doesn't really grasp what you're saying about this. And I said this to Rob when we were emailing. I was like, I feel like I'm worried that you're not getting what the point is, because even though Dan and I, well, I'll speak for myself because I don't know where you were at on this. Despite the fact that we barely touched on the paper in the first video, and in the second paper, we went a little bit more in depth, mostly arguing with Rob, and I could have been more clear on this, that infinitesimal p-value is not coming from this, starting point, because Rob's argument is basically that they're starting with random character states, and then that's a straw man of creationism because God could have created with common design. So some of that low p-value is erroneous and shouldn't be included. And I guess we're going to get to this, but what I'm trying to say is that it's like Rob is very interested in that singular particular point.
Literally one line.
Yeah, it didn't have anything to do with the argument that we were making initially. It didn't have anything to do with the argument that we're making the second time. And it doesn't have anything to do with the argument that we're making now. But now we are empowered with the actual nitty-gritty methodology of the paper. And so are you. So let's enjoy what Rob has to say.
Let's do it. Okay. So I'm going to play the first, seriously, 5 seconds here. And I want everyone to just put a pin in that. Just put a pin in that for later. Okay. And then I'm going to jump a few minutes because we don't have to do the 4-minute review of everything that's happened. But we're going to just first few seconds I want to get, though.
Nothing kills good science more than hidden assumptions.
That right there. Put a pin in that line. We're going to need that later. Just be thinking about that. So now we can jump up. We're going to play only about 35 seconds. We're going to go up here to about 4:04 is what we want to do here. Okay. Here's what we're going to do. So this is Rob talking about this paper, and we're just going to talk about this one little bit. This will take a couple minutes. It's about 30 seconds to him. It'll only take us a couple minutes, I think. Here we go.
To cut down the forest because it was a straw man argument. And they also put a lot of emphasis on this particular manuscript. Maybe you'll remember And this manuscript saying that there's strong statistical evidence in favor of common ancestry or the single tree of life. This paper also, when you read the methods carefully, there's this big assumption, it's a huge straw man assumption, that each family ancestor at its origin acquired character states at each character independently of all other family ancestors. So again, they're assuming no correlation structure at the beginning of life.
Okay. Now, I want everybody watching, whether you're watching live or in the future, hello, future viewers, thank you for being here. Think about what we just heard from Rob in the context of what we just went through, right? Because Baum, in the paper, right, says, here's how we did our simulations, right? And now Rob is taking that to mean something that it does not mean, right? Baum et al. Did a Monte Carlo simulation. They're randomly sampling to build a distribution. So when Rob goes, oh, did they have random state? Yeah, they randomized their data. That's what their methodology was. That does not mean that the optimal starting states were random in terms of different from each other or uncorrelated with each other in some way. They were uncorrelated in that one did not determine another one, but there could be, and I would assume would be, a hierarchical pattern of similarity in those optimal trees, right? But that sentence that Rob plucks out there does not mean what he says it means.
To be clear, right? Like, it takes a little bit of background for phylogenetics to be able to dig into some of this stuff. Like, I fell into the same trap on a cursory look because the last time I had read this paper all the way through was, gosh, years ago now. So it's like, it's an easy trap to fall into. But the problem is, like, there's definitely a difference between like taking a cursory pass at it and being like, oh, I think it says this, right? And then having, a video dedicated to the paper itself and misunderstanding it in that context, right? Because that's a choice. Like, making a video on something is a choice. And I would hope that if you make an entire video on it, you would have a decent enough understanding at that point to confidently say what you're saying moving forward. But like, Rob seems to think, to be abundantly clear, that the issue with this paper, the issue with Baum et al., is that it doesn't take into account that God may have designed things with common archetypes in mind. So the idea is that God has to, this is what Rob thinks the paper did, that the paper started with a completely random state for baboons and their closest relatives, the cercopithecids, and then a completely separate, uncorrelated at all random set of states for hylobatids, like the gibbons, and for hominids, so the apes, excluding humans, obviously, of course. And so his thinking is that, well, that's erroneous because God designed, monkeys and non-human apes to both have 5 fingers on every hand and the same number of the same basic dental formula, and they all have hair, and they all, have relatively long lifespans, and like all of this kind of stuff. So it's like he's thinking that that's where those low p-values are coming from. It's not.
It's not.
Because they didn't pick a starting state and evolve it forward. That's not the methods of the paper. And I understand that now.
Wait,
poor Rob. We could have just gone on our merry way, and I wouldn't have known the details of the paper, and you wouldn't have had to do a deep dive. But now we're well armed on this subject.
Yeah, and to be clear, the authors did what Rob wants them to have done, right? They probabilistically determined what the best case scenario is, what the most parsimonious configuration is for the separate ancestry hypothesis. They didn't do that by picking specific starting points that they think were good. They let the data talk and determined probabilistically what those optimal starting states would look like, right? And then the comparison they did was to show that there's no way, given the separate origins, to get the level of parsimony you get with the real data, right? That's the problem that Rob has to deal with. But the comparison is not based on just any old thing. It's based on probabilistically determining what the best case scenario is for the separate ancestry hypothesis, and then showing that the actual data deviate, let's say, significantly from that best case scenario in terms of parsimony, right? So there's kind of two ways that we can respond to Rob here, right? The simplest one is to say he didn't understand what was going on, right? He misinterpreted what this is. He's not an evolutionary biologist, not an expert in phylogenetics. He misinterpreted what was meant, and it's actually what we just described, and that's the basis for his misunderstanding and why he's making the argument he is, and that's that. But we can actually be more generous to Rob here. We can assume that he's making a more sophisticated argument than that, right? So here is my attempt to like steelman Dr. Stadler as hard as I can. And Rob, if you see this, let me know if this is what you're actually going for, right? So this is the most generous interpretation that I can come up with here, right? His critique is that the randomized data are not generating a realistic distribution for the separate ancestry hypothesis, because the actual process of creation of these separately created groups would either, we got two options here, right? Either A, it's going to result in a stronger artificial phylogenetic signal, i.e. shorter trees, right? The distribution was wrong because it didn't find trees that it should have found, right? That's one possible critique here. That distribution, for whatever reason, did not find the optimally short trees. Or he could say the process of creation would have involved hierarchically correlated family ancestors, meaning that the distribution that we actually got was artificially wide, because some of those trees in that distribution are going to be highly correlated in those family ancestors, and some of them are going to be quite different, right? At, you know, the since we're using families, I don't know what some families and primates would be, but like some of those trees are going to have them just by chance very different from each other, and that's going to be a very non-parsimonious tree, right? So he could say we shouldn't be doing the statistical comparison between the whole distribution. We should only concern ourselves with the most optimal fraction of that distribution, right? That is a critique he could be making here. I don't know if that is what he is intending, but that is, I think, the optimal kind of interpretation of what he is saying.
So I have a different. I think Rob's going to go in a very specific way.
Can we put a pin in that? Don't forget that, because I want to go through my two options here, A and B.
Got it.
Make sure you write it down. Don't forget it, because I want to come back to it.
I'll type it in my phone.
Now, option A that I said, right? His critique is that the process of creation would have resulted in a stronger artificial signal than we actually get, right? That is begging the question. And I'm using that in the correct sense. He is assuming the conclusion he wants, saying that distribution is wrong. There are actually data points that are part of that distribution that you didn't find, right? And if that's the case, then I invite Rob to show that that's the case computationally. Pick up the baton, you have all the methods, generate your own distribution, and show that the range, the minimum on that range that we just talked about, should be a lot lower, right? Show that that's the case. You can do that. Publish that paper. That'd be awesome. Like, show us that that's the case.
Yeah, right. So to be clear, though, that's the ball is in his court on that, because at present, conventional science has tested the best version, as we understand it, of family level separate ancestry as a scientific concept.
Yes.
Right? So he's responsible for it at that point. He's got to say, no, actually, that's not the best version of family level separate ancestry. Here's how you could have improved the comparison. Here's how I did it. Here's why it's better. Like, you have to justify that typically in science. Like, it's not good enough.
So
in
I'm going
to
to. So I'm going to clear a bunch of this stuff, and we're gonna visually do this. Okay, we're gonna visually do this because I think it will be useful to illustrate this visually for everybody. Okay, what I'm actually saying here, right? So what we're saying is, we've got our distribution, right? Whatever it is, we have our distribution of randomized data and real data, and they don't overlap. And either Rob is saying, well, this distribution should be wider, right? And if that's the case, rerun it, show us that the distribution is actually wider. Or he's saying, this is wrong. You have to only consider the good stuff. You can't consider this. You should only be doing your comparison between the optimal trees, because that's how it would have been created, right? You can't consider this. This is not how a designer would work. You need to just use the best and compare it to your actual distribution, right? And for that second critique, the first critique, he's begging the question, and run the simulation and show us that the distribution is wider. For the second critique, he can actually show that that's right. Because what you can do is actually, you can concatenate that data set, right? You can just cut it off at some point and rerun the analysis. But here's the thing, the data sets don't overlap with each other, right?
Yeah.
So you can do it where you can basically reverse the common ancestry test, where you're doing the real distribution for family separate ancestry and the single best tree right there. The single best one. And you could say, is this tree part of this distribution? You could basically flip the directionality of the comparison to make it the opposite of the common ancestry test I drew in purple earlier and say, there it is. Now, the numbers for that are not great for Rob, because remember, the real range of lengths over here, the minimum is 11,000 something and the maximum is 12,000 something. And this minimum right here is 16,000 something.
Yeah.
But hey,
thousands off.
Run the test, see what the p-value is. Like, that is a thing Rob could do. If his argument is the version that I just articulated, run that test and show us. And again, like Erica said, ball's in your court. This is a published paper in the literature. It stands, on its own. If you got a problem with it, pick up the baton, do the analysis, and show that it's wrong.
But like we just saw visually, or like you could see in the table that was on the screen moments ago, the p-value's not going to be great. Like, is it going to be better than the p-value as it stands? Like, I don't know what the point would be of doing an exercise like this, but by all means, I think his best bet would be, if you want to keep things scientific, which again, asterisk, you're going to have to find some kind of flaw with Baum's methodology here, because the straw man thing doesn't hold any water. Yeah. And it's got to be, at least insofar as I can tell. I mean, you're more astute with this topic than I am, Dan, more formally educated in it, to be sure. So it's like you would probably be capable of picking out his potential responses than I would be. So it's like, that would be a good starting point, is to go with the two that.
Let's see. Right? Let's see. Let's just sit and look, Rob, even if that wasn't what you were thinking, you can come back and say, yeah, that's what I was thinking. Let's do that, right? Go for it. Knock yourself out, right? Like I said, I think the simplest and the most likely explanation is he just didn't understand the methods, because he's never professionally studied phylogenetics. Like, it took me, the better part of a week to get back into this paper, having not done it since grad school, right? And this is stuff that I'm very familiar with from grad school. And I had to really, get those muscles back, right? Somebody jumping into it cold, I think the most likely explanation is that they're not understanding the methods. But I've offered a couple of alternatives that are more substantive critiques.
And it would be like, there's no shame in not understanding that's something that's not your field. Like, I'll be the first person to say, if Dan hadn't gone through all of this stuff and put up with me asking him questions over WhatsApp on okay, what do you mean when you say this, right? I could have fallen into the same exact trap as Rob did, because I hadn't read the paper through freshly the first time. I did fall into the same trap. But there's, again, a very big difference between making a passing comment on a paper and making an entire video sort of tooting your horn that he's got us red-handed, Dan. He caught us in a huge mistake. And it's like, no, not quite.
So we're going to jump ahead a little bit. We're going to play about a minute of the video. So bear with us, everybody. But this is where emails back and forth with the author, David Baum. And Rob is going to lift a line out of one of those emails. And we're going to talk about how he's misinterpreting that line as well. And you'll be able to see in the words, given what we talked about, exactly what David Baum means, and then how Rob is misinterpreting those words. You're gonna see it, and I bet a bunch of you in the chat right now, you'll be able to go, I see exactly what's going on here. So again, bear with us. This is about a minute long, and then we're going to come back and talk about this. And after that, I promise you we're close to the end. We're almost through all of this, because there's a lot in here that is not relevant. We're going to jump through it and just kind of touch. So here we go.
In the meantime, when all this was going on, we've had some correspondence with the study authors. The study first author, David Baum, we wrote to him, and I asked him specifically about this assumption As with species-separate ancestry, we assume that each family ancestor at its origin acquired character states at each character independently of all other family ancestors. And I asked him to clarify that. And here is a quote from what he wrote back. He said, "The family separate ancestry test assumes that each family originates with a random character state. Two families could get the same state for a particular character, but this would not change the probability of those two families sharing the same state for another character." In other words, that's the definition of random, and there's zero correlation, zero hierarchical structure then given the designer to start off life. And
Okay. That last little bit is where, like, he was fine, and then he drew the conclusion that he wanted to draw that was completely wrong. Because what's going on here is David Bohm is describing that random sampling technique that we talked about, right? The Monte Carlo method, where we're randomizing the data and we're just plucking optimal phylogenetic trees. When we randomize the data, each character is going to be assigned, or I should say each character state is going to be randomly assigned to a taxon independent of other character states for that same taxon, right? So having an A in your DNA at one site doesn't tell you anything about what you have in another site, right? They're independently assigned and independent of the characters assigned to that state in the other taxa, right? That's what a random permutation of the data is. Bohm, I'm not in this man's head. I did not directly correspond with him, but if it was me, I would be like, why are you asking me to repeat the words that I wrote in the paper? I wrote the words in the paper. I'm going to repeat them to you now. They're right there. That's it.
So Dan, if I may, someone in the comments, so Eric Massa, no, is it Eric Massa? Yeah, Eric Massa in the comments asks, do you and or Erica plan on contacting Dr. Bohm, assuming you haven't already?
Oh.
Why, yes.
Funny you should ask that.
Yeah, it's funny you should ask that. I mean, I'm gonna have to go through Stadler's video with a fine-tooth comb another time, because even outside of the Bohm stuff, there's a bunch of other stuff in there that we could just sit and talk about for ages. But it's like, yeah. And hilariously enough, right? Like when Bohm responded to me, and I'm gonna get into this a little bit more. I'm not gonna show any emails by anybody who hasn't given me.
Hey, Erica, I apologize for interrupting you. Did we skip what you had noted on your phone a minute ago?
No, I imagine we're coming to that later.
Perfect. Okay, great.
Because it's what you you already know it, Dan.
We'll get there. Great, perfect.
Anyways, what I was going to say is that, and I, I'm not going to share any emails of anybody who hasn't given me explicit permission, but it's like when I spoke with David Bohm about this, he was very tickled by the fact that so many people were emailing him and asking him about this paper.
It's like multiple people.
Yeah, and at one point he was so if like creationists like stumbled across this paper, like are they trying to deal with it again, like as if he's been through this rodeo before? But to give full transparency, right? I saw the same email that Rob saw because we were in the same email chain, and so when I saw Bohm's response, I was like, oh, okay. I was like, okay, so I guess is it possible like that he started with random states? But I was like, no, I don't think so, because there's this weird thing that creationists do where when they get sort of an inkling about something in a field that isn't their own, evidently their first thought, at least some of the time, and we saw a little bit of this with Stadler, is that it's like, the authors must be wrong. They must not have considered this super obvious issue that starting with random states and assuming no correlation and evolving forward.
It's a Jensen move. It's telling the authors they're wrong. Yeah,
it's a Jensen move. It's telling that it's sort of implying that you know better than the authors, that the authors couldn't have possibly considered this. And I'll say, as somebody who's been humbled many, many times on YouTube and also in my academic career, my first thought when I read that was, okay, so I'm definitely missing something. I am not understanding what he's done. And that's when Dan and I started concocting, like, okay, we need to do a deep dive on this methods, because clearly there's something going on here where and I'll speak for myself again on this point. I'm missing something here as to what they did. But in a sense, and I have to emphasize this because I emphasize it to Dan and I emphasize it to Rob too. In a sense, that shouldn't matter as much. And the reason I say it, like, it's always best practice to know the fine-tooth details of every methodology of every paper that you read. But that is not a realistic expectation for every scientist in every field to be able to do in fields that are not their own. That, you guys, is the purpose of an abstract. It's the purpose of an introduction. It's the purpose of peer review, right? So that you can get an idea and trust that the results have been filtered through several different other people who are experts in the field, so that you can look at this and say, okay, maybe I don't understand the details of the methodology, and I should, but at least the results, it's not like a moron sat down and did this, right? Like, these are people who are like forerunners in their field.
They probably have thought through the obvious pitfalls of the methodology, right?
Right.
Yeah.
So, another, Gutsy Gibbon learns another painful lesson where it's like, okay, it turns out don't talk about any paper unless you've emailed the authors and figured out exactly what statistics they did.
Here's a paragraph. Is this an appropriate way to describe your paper? And once they say yes, then you can say it on video, right? I mean, Erica, we've done this, and again, peek behind the curtain. We were talking about this before this Stadler video came out, and then the Stadler video came out, and we were like, here we go. But this is also gone in the other direction, where I've workshopped stuff talking about, like with the recent Casey Luskin stuff, with the genome stuff. I've run my stuff past you to be like, is this the appropriate way to say that? Oh, it's not the appropriate way to say that. What's the right way to say it? Why was that wrong? Right? This is what the process is when you're operating outside of your field. I'm not a paleoanthropologist. I'm not an expert in human-chimp genome comparisons. Erica had to explain it to me very slowly, so I didn't say something stupid in my recent videos on the topic, right? That's how this works.
She's being very generous. Certainly, I would say that I am close to an expert in the area that I'm actually getting my doctorate in, but I have had to learn a ton of genetics for the doctorate itself, and then also for all of these shenanigans on YouTube. But the critical difference is Dan and I aren't trying to overturn foundational concepts in biology by misunderstanding a paper, right? When we misunderstand a paper, we're like, oops, my bad. Here's the actual reality of the paper. And I guess by happenstance here, and I don't know, I don't think we were even abundantly clear. Like, I don't think either. I mean, I'll speak for myself again. I wasn't really 100% sure of the detailed minutia of the methodology in the first place. But then again, I have a lot of trust in the authors of most papers to summarize it for me in the abstract. But one thing I can say for certain is that I didn't make, a video saying that this paper does not say actually what it's saying it says, and that the author's actually wrong because they didn't take into account like a really basic like a very basic concept in their own field as someone who is outside of that field.
Yeah. Agree. I co-sign all of that. Yes. So I want to show this comment right here. This is a really useful reminder. Dan, could you emphasize that this process of assigning independent characters is done many times to generate the Monte Carlo distribution? Yes, this is the key thing that we should keep harping back on. And in fact, I want to talk about this a little bit more relative to what Rob just said, because he basically, they're pulling random characters, doing the comparison. That's not representative of the separate ancestry design hypothesis, right? But again, the pulling the random characters and assigning the random character states was to build the distribution of tree lengths from shortest to longest, right? What the comparison we want to do is we want to compare the optimal trees from the real data, right? The most parsimonious real trees to all those most parsimonious trees with the randomized data. The trees made from the real data are just what they are, right? You're going to tell us that real data are going to tell us what the parsimonious solution is, what that looks like. The point of doing the Monte Carlo simulation is to find the shortest Markov chains from ancestors, plural in this case, to all the descendants. Now we have the extant sequences, right? So that's locked in. We don't have what the starting states are, but we're probabilistically figuring out what that optimal pathway looks like, right? They're random in that we're not determining what those character states are going to be, what those common ancestors, either at the nodes or for the whole family, are going to look like. But they're ultimately, if I had to guess, if we were to like dig in and like actually pull up that minimum length run, I bet they are highly correlated with each other. They're probably exactly what Rob wants them to be and expects them to be, because that's how, just like intuitively, right, that's how you would get the shortest possible tree, is you would start with states that are correlated in a hierarchical way, minimizing the number of convergent mutations you need along each of the actual branches that are evolving on your tree. right? They're trying to find the shortest trees. They're going to have the optimal starting states. That's the point of the methodology, is to do what Rob is saying they didn't do. So again, I think the most reasonable conclusion here is that Stadler and also James Tour, because this is kind of on behalf of James Tour, right? They're unfamiliar with the techniques. They're hearing these words, they're reading these words, and then kind of mapping their own desired conclusion onto them, independent of how the actual methods work. I think that's what's going on here.
If I had to guess, I would guess that that's this. I would guess the same. I would guess that would be the interpretation of Rob's understanding. I would take the same interpretation of Rob's understanding of the paper. And the reason I say that is because it seems like in this video he said as much when he was talking about the quote and then elaborating on what he thinks that quote means. And I think you could only get to that point if you didn't know what these methods actually like functionally did, right? And again, like there's no shame in that unless you're claiming that the author's paper was pointless and didn't test what it says it tested.
Yeah, and I just want to highlight this comment because this is also a pet peeve of mine. The misuse of the word random in creationist circles is infuriating. That's from Misinterpretations, which is a fantastic username, A+. This is why I explicitly avoid the word random unless it's explicitly correct, as it is like in this case when we're doing a randomization technique, right? But like talking about mutations, for example, I use the word probabilistic, not random, because probabilistic implies it's the opposite of deterministic, whereas random, people hear random and they think coin flip, roll of a die, every outcome is equally likely. That's not the case, right? You get a random distribution where certain outcomes are more likely than others. So I like to use probabilistic as a more precise way to describe that doesn't give the false impression of a flat distribution, right? I don't like the word random, and creationists always mess it up. It's very frustrating.
I think which we've just said covered Sharon's super chat for $2 where they say, I'm lost. It sounds random. I think we covered that, but do you want to add anything to that?
I believe we covered that. And also, thank you. It's based on how we're generating the distributions probabilistically, right? As a basis for comparison. We are randomly generating what those most parsimonious trees might look like. And then we're comparing it to the actual tree and saying, is there a phylogenetic signal or not, right? And even in the family separate ancestry, the answer is there is. And
to be clear, for Sharon here, right, Rob's issue isn't necessarily with the nature of randomness in this experiment, in these methods. It's randomness at the starting point, which is then evolved forwards, uncorrelated, which he thinks is generating those erroneously low p-values. That is not what's going on here.
That is right? Not happening. Correct.
It's not randomness in isolation. It's randomness with random starting points that are then sort of let loose to run under normal
level. It's random sampling from a distribution.
Right.
And and that's the thing. Try to explain, you have to simplify the phrase random sampling from a distribution. That's kind of minimal. That's what it is. It's just what it is.
Right.
Yeah.
Right.
Okay. So I want to play just a short little bit here, just a short little snippet that we can all just appreciate. We don't even have to respond. I just want to appreciate the irony in the approximately 6 seconds I'm about to play. I want to make sure I'm at the right time point. Yeah. Yes, good.
That, which are wrong. They are misleading all of us, and they have misunderstood the entire paper. And all of this is...
They're misleading us.
I feel like it's because Rob doesn't understand the entire paper, Dan. Right?
I feel like he's misleading his audience, and it's because he doesn't understand the entire paper. Rob, I apologize, but seriously, that broke my irony meter right there. That just shattered it into a million pieces.
Well, and misleading doesn't have to be intentional. I don't think, we both dealt with malicious creationists. I've dealt with people who I think are intentionally lying. I don't think Rob is intentionally lying. I don't think misleading and lying are synonymous necessarily. I think it's possible to not understand something well enough and accidentally mislead your audience, which is what clearly happened here.
Yeah. So let's play. It's not quite a minute. It's about 45 seconds here, and this is kind of the end of the section on this paper. And then I just want to play. And I know we've been going for over 2 hours, Erica. How are you doing?
I'm okay. We should keep going. Let's knock it out.
Let's knock it out. We're gonna get through. It's just jumping at this point. So let's do this 45 seconds, and then we'll go from there.
An argument. Now, I suspect that Erica and Dan are going to respond to what I'm just saying here. They're not going to give up, and they'll be claiming that the paper really is all about the hierarchical structure that's present in differences between families, and that all that evolved after life began. But Erica and Dan, with this assumption the paper clearly makes, without any doubt, that they're starting with randomness at the origin, then any kind of hierarchical structure seen in modern life, any kind, whether it's a hierarchy of differences or hierarchy of similarities, it's going to favor strongly the tree of life over the forest of life for all the wrong reasons, because they assumed a certain kind of designer that we all know no one who designs anything ever does what they're assuming.
And then we get into one of the James Tour infomercials. Erica, you clearly have thoughts.
That sucks, dude. Like I've been there. There's nothing worse than being on camera and just saying something that is just blatantly wrong. Like and then the editor assumes random at origin in text on the screen. That sucks. I empathize with Rob on this one. That's not
spike a football super hard only to realize you ran into the wrong end zone.
Yep. That sucks.
Rough.
Like I feel like I feel empathy for him on that. But then there's another more vindictive, more petty ape-like side of me that sees that, and I'm like, I warned you, dude. Like I warned you on this one. I was like, make sure we both did. We were both like, make sure you've read the methods really carefully before you make a video on this. You could have pulled back. Like, I know he got those messages before this video came out, so it's like he could have pulled back and double checked. Which I would love. Can you imagine if someone gave you a warning every time they knew you were going to get something wrong and you had the opportunity
that would be amazing.
to double check yourself? Like,
yeah,
I would love for that to happen.
Yeah, it's like the trap finder benefit in Dungeon Crawler Carl. It's like, whoop, watch out, don't step there. Like, hey,
don't go there.
Yeah.
But like,
I just want to take a moment to thank Zachary Weatherford for gifting 20 Creationists memory. Oh my goodness, thank you. Thank you. That's amazing. Thank you. Okay, so you were saying.
Unambiguously, right? Like, based off of what we've gone over today, based off of my now much better understanding of the deep methods of this paper. And the irony is it's not even that deep. It's just the depth on one type of the methods that for one specific of the tests, of the many tests, as you said, that they ran. It's like, it's abundantly clear that Rob doesn't know what they actually did in the paper. He doesn't know. That line says it all. Assumes random state at origin, not what they did.
It's talking about generating the distribution. Right.
Right. It's based on a misunderstanding of bomb in the paper and in an email sent directly to him.
Yeah.
And you know what? I'll add that, like, the email was longer than the bit that Rob shared, and a previous email was longer too. And so it's like, I think that the clarity in that email potentially makes it worse because the bomb is very clear that it's like, if you are a creationist, this paper hurts you deeply, and no, it's not assuming zero correlation at the starting point and then that deep say, like, that's just not anything to do with what the paper actually did.
Yeah. So Erica, here's how I want to kind of wind this down for us. There are a couple of things I want to show here. There's one specific 30-second clip I want to show, and I want to juxtapose that with some earlier stuff that Rob has said about us mischaracterizing his arguments. But what goes on here now, he changes gears. He's done with the bomb paper. He spends a big chunk of this video then on you specifically. And as I understand it, I don't want to give you homework, but as I understand it, you're going to cover that. You're gonna give it the Erica treatment.
Don't worry about it, Dan. You can skip that stuff. I'm planning on taking a fine-tooth comb to it, as only I am cursed to do.
And then he comes back to my little simulation game thing that I did. I am going to cover that separately because oh boy, he did not follow what I did. To the point where he shows like it's this percent hierarchical and this percent random, and it's just wrong. Like what he shows, the numbers are just wrong. So we're not going to go through that here, but that's in this. I will cover it separately.
I'll add that I tried to explain that in our email conversation, like to the best of my ability. I think I did a decent job, but it was a layperson understanding all the same. And so I was like, we can rope Dan in on this if you want.
I can help. Yeah, no, it's just like he got the numbers wrong. Like he just didn't understand what I did. And so I'm going to cover that. And then he has a bit on Winston Ewart's dependency graph thing, which I'm also going to cover separately. I've been working on that, actually digging through those not papers. Rob does it's a real journal thing. It's biocomplex. It's not real journal. It's fake journal. Like we're not having that conveversation. So I'm gonna.
The ARJ is also a real journal because they have intertelling peer review of paper on genetics, right? Like it's not. We're not talking about the rigor that is required in conventional science. That doesn't mean creationists can't publish in regular journals. On rare occasions, they have. So it's put the dependency graph in a normal journal if you think it's up to.
Yeah, do it for real. So I'm going to cover, so I've got 2 pieces of homework that I have to cover separately from this. Erica has one that she's going to cover separately. But before we end this, we have to show 2 final clips here, okay? So the first clip is we're going to go to 12:29, and we're going to play for about 30 seconds. And, folks, after this, I'm going to show a little compilation. I'm going to put a little montage together. We're going to have fun with this. So
well, you're finding that none of you, well, some of you out there might know, but the wonder of being able to see unflattering like stills of you caught mid-sentence is an endless source of joy for me.
Yeah. I agree with that also. All right, so enjoy these 30 seconds, everybody. Then we're going to put up a different video, and trust me, you're going to appreciate it.
And you can directly measure it and you can prospectively study it. But to see if humans and bacteria share genes from a common ancestor, meaning you're giving a reason why they have common genes, that they came from a common ancestor. In order to prove that with confidence, you've got to find that common ancestor and you've got to watch it evolve prospectively, directly looking at what it's doing, handing off genes to bacteria, handing off similar genes to an evolved human that comes from the same lineage. And then you've got very convincing evidence. I hope everybody can see there's quite a dramatic difference there.
So you have to directly see it in order for it to be high confidence evidence, right, Erica? I think that was pretty clear, that you have to watch a bacterium evolve into a mammal to have confidence that that is a thing that happened, right?
He even says prospective study. He couches it in perspective. So looking forward, right? Prospectively studying it, and then especially playing the little March of Progress clip. But it's like, maybe whatever. Maybe we're misconstruing him here, but Rob keeps Baileying this.
Well, Erica, let's see if we're misconstruing him all the way back in an earlier video in this series, because I went back and I put together a little montage for us.
Did you see?
Okay. Now, this is, we're going to see 2 clips from an earlier Stadler and Tor video where they're making fun of us for saying what that Stadler says you have to see bacteria evolve into mammals, right? So here is this video. This little bit is from the very beginning, the little trailer part, and then it's going to jump to a clip from that, and then we're going to see Rob's, what we just saw. So let's just see, everyone, as Rob says, see if you can spot the difference.
Were you there? Did you see all of history?
The directly observable criteria here, this one might be a little more subtle. And I'm not trying to say here that you literally have to see it with your own eyes, although that's probably the most confident of the results, right? But the more directly you measure a result, the greater the confidence you give. But the people who have rebutted us took this to kind of an extreme, and I'm afraid other people might do that as well.
He falls into this trap. It's literally if a person doesn't actually physically see it with their eyes, it doesn't count. And like, okay, great, congratulations. Like, toss out paleontology, astronomy, archaeology.
Okay, you can see what happens when you take that to an extreme. But to see if humans and bacteria share genes from a common ancestor, meaning you're giving a reason why they have common genes, that they came from a common ancestor. In order to prove that with confidence, You've got to find that common ancestor, and you've got to watch it evolve prospectively, directly looking at what it's doing, handing off genes to bacteria, handing off similar genes to an evolved human that comes from the same lineage. And then you've got very convincing evidence. I hope everybody can see there's quite a dramatic difference there.
I don't even know what to do with that.
What?
Like, seeing it side by side like that actually does hit so much harder, because I forgot he was straight up like, that's not what I'm saying. And then, Dr. Stadler, why?
What do I do with this? What do I do? I don't know how to respond to that.
Okay. Rob, do you or do you not think that in order to provide high confidence science for evolution from a last universal common ancestor to extant life, do you need to see all of it to provide high confidence support for it? Like in a prospective study, like with your eyes, as you said?
Or can we make accurate predictions based on extant, directly observable data about what must have been the case? Does that count? Like, that clip, when I saw that, when I watched this video for the first time, I stopped, I took the timestamp, I was like, that is amazing.
That's actually, that's like probably calling your pants down, dude. Like, that's Rob.
Put that in a museum.
Why didn't you watch your previous video first? I don't know if you do this, Dan, but sometimes when I'm responding to people and it's like a back and forth, I'll like watch my previous version.
What did I say?
Because I'm like, what did I say? Just to make sure that I don't like contradict myself accidentally, like that I'm accounting for things that I knew at the time. But this doesn't fall into that category, right? Because this is something that is just it's like a logical thing.
We and Dave were making fun of him for saying you need to directly see it. And then he's like, that's not what I'm saying. You don't need to directly watch evolution happen with your eyes. Don't be silly. I wasn't saying that. And then he comes back and says, you have to directly watch the bacterium evolve into a mammal.
And we were even like a little sheepish in the second video. We were like, all right, we probably shouldn't have, gotten that badly on it.
We were right.
Yeah, that would be an insane thing to say. A crazy person would have to say that. Like, we weren't giving him enough credit. Like, Rob,
well, here we are.
We're getting your goat a little bit here, but like I think your goat deserves getting got. I
think we earned that one. Okay.
For some reason, and I honestly
be quiet. So the last thing I'm going to do, I'm going to jump right to the end here because again, there's a whole bunch of stuff in there that we're going to deal with separately. But I want to go up to about the 37-minute mark because he's gonna end this conversation, as you'll see. But he earlier predicted how we would respond. And I do want to point out that his prediction of how we would respond was wrong. Which is in contrast to how I predicted he would make this video. I predicted two specific things he would say in this video, and I nailed both of them almost to the word. So that is gold standard, folks, confirmed predictions. And then Rob says, here's how Erica and Dan are going to respond. And we did not. Instead, we spent 2 hours going through the methods of the paper, was how we did it instead. So let's see.
That's just, yeah, that sums it up.
That's extremely on brand. And we got to it proudly.
It's on brand. But like, that doesn't mean our original point wasn't also true, though. Like the pattern overlaying, like the overarching pattern does still matter. That's just not, I mean, we took it a little overboard, I guess.
So let's just get this last little bit because it is wonderful. And then Erica, before we do this, I want to make sure you said the thing you wanted to say, right? I just want to make sure we don't leave out the thing.
We're going to finish this, right? This is
okay. Yeah, we're just going to play the very end and then we're good.
Let me get my note here then. Here's the problem with Rob, right? And I was going to get into this, I think probably when we were going to touch on some other clips, but we're going to do those separately because we've been going for 2.5 hours. Rob does not have a testable model of separate ancestry or of creationism in general. That's the problem. Consistently, when I was saying earlier that it's like this paper kills the scientific version of separate ancestry, it does. But Rob will never cop to that because Rob's version of separate ancestry isn't scientific. It involves all sorts of like rando classic creationist mechanisms like genetic entropy, created heterozygosity. At one point, I believe he's used this word elsewhere as well, but designed mutability, like all of these ideas that exist in the creationist lore and nowhere else. They aren't found in any scientific literature, so you can't appeal to them unless you have demonstrated that these are real mechanisms at play, which means that until you demonstrate those, and until you demonstrate that there are problems with Baum et al.'s paper here, this paper disproves separate ancestry from a scientific perspective. Full stop. That's it. That's the ball game. If you want to appeal to all of this extra stuff, that's fine. If you want to sit around and try to poke holes in evolution, that's fine too. But as far as a scientific model of separate ancestry, it's DOA at present. And y'all have your work cut out for you on what to do to resuscitate that boy on the beach, right? So that was what my idea of Rob's response or potential understanding of this is that it's like he can't have a scientific model because in his mind it supersedes science. Like he doesn't want to do this methodological naturalism stuff, which, by the way, was in part founded by well-meaning Christians who wanted to understand the world around them. But modern day creationists don't think that that's good, even though their forebears were the ones that seeded some of that during the Enlightenment and onward, right? No, they want to appeal to magic mechanisms. And I know that's like a pejorative term, but that's what it is. Until you can show that this is a mechanism that exists, it's a magic mechanism.
I just want to I'm going to snark a little bit by highlighting this comment from Templar Warden. So he doesn't have a prospective study. Sounds low confidence to me.
That's the problem. That's the problem.
You got to have a way to test it. You got to have a way to test it. And what this study shows, the whole point of this paper is, hey, let's come up with a way to test it. And what they showed very rigorously was that the data as they exist, the extant genome sequences that they used, are incompatible with family separate ancestry, right? That's the takeaway here, right?
Yeah.
So with that in mind, let's see how Rob wraps this up. Let's see what the next installment is going to be.
Job showing us how this works to make science go bad. And with this, folks, I think our discussion, we've had like 5 back and forth videos at this point. I think our discussion has run its course. Unless something revelationary comes up, Dan and Erica, you're welcome to have the last word. But in comparing these two hypotheses, the tree of life and the forest of life, I've been saying that we need to focus on the types of evidence that provide high confidence, things like long-term evolution experiment. And that's where I'm going to go next, is focus on producing a review of all the evidence out there that gives us high confidence and see how that evidence stacks up against tree of life and forest of life to know which one is more correct. So thank you, folks.
Okay, I just want to put a prediction in the ground because you mentioned the Lenski long-term evolution experiment. He's going to do the thing about genome degradation, about how they're like the die-off that was reported. That's what he's going to talk about. And he's wrong, and creationists always get it wrong. Prediction. Hold me accountable, folks.
I just want to say that, like, I had watched this before, at the gym when I was breaking my PB, but I forgot how he ended this. Like, I forgot the exact verbiage of how he ended it, as opposed to, like, or not opposed, but like taking into consideration where he wants to go next. And that's so beautifully underscored what I just said a couple of minutes ago. Rob knows that there's no model. He knows there's no model. Otherwise, he would pivot not to trying to poke holes in evolutionary theory, but coming up with an actual robust model for creationism, any kind. But he can't, because his model requires stuff that doesn't exist. It requires stuff that doesn't exist. Created heterozygosity, genetic entropy, what's the other? A design mutability, like all of
that one is bananas. The designed mutability one is genuinely bananas when you like actually examine how it's supposed to work mechanistically, because you run right up to God is picking which mutations happen is because it can't be probabilistic. You can't set. It's not like dominoes where you set it up and then knock it over and it makes the pattern. Mutations don't work that way. If you're going to design it so a certain set of mutations happen, you have to actively guide those mutations to happen.
Not only that, but like, I hope that the extreme end point of this is clear to everyone. If design mutability is on the table, if created heterozygosity is on the table, if genetic entropy is on the table, again, 3 mechanisms that we have absolutely zero goose egg support for existing at all, then there is no way to tell where design stops and descent begins. Because Rob accepts, for example, that all dog breeds are related, and that dog breeds are related to wolves, and that wolves and all the dog breeds are related to foxes in Canada. He accepts that. That's why he's okay with family level separate ancestry, at least some of the families, right? So how do you tell the difference between descent within those families and design prior to those families? You can't. And because you can't, theoretically, you can take this to last Thursdayism, right? Theoretically, God could have done designed mutability all the way up to the individual level so that all of human relatedness to one another is just an illusion. Your relatedness to your mom and dad is just an illusion. You were created last Thursday as is, and every single baby that's been born since then, that's designed mutability. It's all just a ghost. There's no way to actually interpret anything of substance or any real history at all, because it's all on the table. God could have done it that way. And that's not science. And if you want to believe that, that's fine. But any phantom of credibility goes out the window in the scientific community the second that you're not just opening the door for a crack for miracles, which is already bad enough form, as many theistic evolutionists will tell you. Even old Earth creationists sometimes will tell you that kind of thing. But you're opening it up and saying that the miracle might be in the test tube, right? It doesn't work.
Doesn't work. I think that's a great place to leave this. Like we've said a few times, Rob, the ball is in your court. And to test these, there's a way to come back at this paper and actually test their model. And also, either or both of us are happy to have an on-air or private conversation on this topic with you. You can email either of us and let us know if you want to do that. We are happy to do that. So with that, folks, this has been a journey. Erica, thank you so much for doing this with me. I know that was a lot, everybody, but I had a genuinely great time going through this paper and figuring it out and figuring out how to convey it to everybody. And it's just such a cool study design to just see how they made it work. I think it's really cool. So I hope everyone enjoyed that as much as we did.
The thing is that I know it's gratuitous, and people are tentative to invite me on sometimes because I can't shut the hell up, and that's true. That's true. But on some subjects, Dan also can't shut the hell up. So it's a match made in hell.
And this is the overlap of that Venn diagram right here, and that's why we've been going. And that's why we've been going for 2.5 hours.
But here's the thing: at the end of the day, that's how science actually happens, right? It doesn't happen with quips on YouTube. It happens with this kind of thing, but done by people who are actually in the field, in the literature, in peer review that happens over the course of years, replicating methods, digging down deep into the methodology. That's where the meat of this stuff is. It's truly exciting. It's a wonderful time to be interested in science.
It's great.
But if you want to get to the meat of this stuff, sometimes you got to put the time in. I know that sucks. We live in a time where it's we all want fast paced, soundbite, quippy. You can't put this shit in a TikTok, is what I'm trying to say. We can't talk about MCMCs in a TikTok.
Yeah. Explain. If you can explain a Markov chain Monte Carlo technique in a TikTok, you are a better communicator than I am. It's I can't do it. Can't do it. Okay. So Erica, what do you have coming up? You always have fun things. What are you doing?
I got a decent. I've got a video I'm very excited about. I've had a lot of creationism videos in quick succession on my channel. So I've got a video coming out pretty soon, hopefully within the next day or so, on the curse of Tomai, which is a really interesting fossil that seems to have bad luck following it around everywhere, in the sense that everybody who studies it goes mad. It's interesting. Sahelanthropus chilensis, for those of you who are familiar with hominin evolution. It's sourced heavily from this recent Guardian op-ed that was just insanely good, that interviewed all the people who are involved. And what's cool about the video and about that article is that it's Dan probably knows this, but when you're in a field, you get all the juicy scoop about people who are also in the field that doesn't obviously make it into papers. You hear about it at conferences or at conversations. So a lot of what's in this Guardian article, I've known a little bit about for years, but it's not something you can really talk about because a lot of it is hearsay, and you don't want to ruin someone's reputation for nothing. But Scott Sayer came with receipts, so I get to talk about this, and it's gonna be cool.
That's awesome. So I got a bunch of stuff coming up. I keep having to move my schedule around because I keep coming up with things to do. And you may have noticed I've started like semi-regularly doing like short little videos outside of my normal Wednesday night time. So what I've got coming up, I'm next week going to show another installment. I can't believe it's been going this long, in my ongoing conversation with Rebecca on this topic, talking about nested hierarchies and stuff. There was a stream that I was on with her and our friend Grayson, and that was a really interesting conversation for various reasons. So I'm going to show that.
Rebecca is such a sweet person. I love that she's very sweet in a lot of ways, truly. But she has some of the most motivated skepticism towards evolutionary theories I've ever seen.
Yes. And I don't know if you watched this one where it was me and Grayson.
I have not.
Okay,
seen
so there was a moment that made both of us go, huh. So it was interesting.
Okay.
Pigs Can Fly was also in it, and he was annoying, so I had to like cut out bits of it because he was being annoying.
I forgot about that guy.
Yeah,
oh wow.
And then sorry folks, this is just us bantering now. This is just an aside, but in the stream I mentally confused him with Bubblegum Gun, and I felt so bad because that is
that's
so
mean.
That's mean. That's
mean.
I was like, wait a minute, are you? No, there's somebody else. I'm so sorry for thinking that. It was, yeah. Anyway, I'm gonna show that next week. You can find it on one of Rebecca's channels. I forget which one, but you can find it there if you want. But I'm gonna show it next week. And then after that, I got to get the schedule worked out, but I'm going to do another of the Genesis Apologetics Conference because Randy Galuza is next.
Oh, Randy.
And Erica, I haven't reached out to you about this, but I'm going to enlist you to come back here because we need to do the Chris Roop talk from that conference.
Oh, I love talking about Chris Roop. Let's do it.
That one, we're going to flip it, right? And then you're going to get to explain it all to me because I am a dum-dum when it comes to that stuff.
Oh my God, I know he's doing the Lucy's a Pygmy thing, so we're going to talk about biomechanics. We're going to get to talk about the minutiae of the lumps and bumps on the pelvic girdle and how we know. Oh, it's going to be great. I'm going to come up with figures.
So we're going to do this again, and we're going to do the Roop talk with the Lucy is a Pygmy thing.
Beautiful, love it.
And then I also have in the works dependency graph stuff. I know everyone's been clamoring for dependency graphs. Give them the dependency graph content and
what they want.
Give the people what they want. They're clamoring for Winston Eward's dependency graph model. Give the people what they want. So I have something on that in the works as well. Plus, I swear to you folks, the stuff that we didn't show in this Rob Stadler video, I'm going to chop it all up and I'm going to show that stuff because you can't just let it go.
You need to know that Dan and I were talking before this. We were like, how long do we think it's going to take? We were like, probably 2 hours total. It was 2.5 hours and we didn't get through half. Like we had to cut it short.
We were just, in my outline, we have everything.
We have everything.
We just cut out about the last third. Yeah.
Oh, well,
folks, this has been so much fun. Thank you all for being here. I am astonished with the size of the audience. It was over 150 people watching us explain how a Markov chain Monte Carlo works. I just love every one of you nerds that was here sharing this experience with us. That was great. Erica, thank you so much for doing this. I thought this was awesome. We're going to leave it there, folks. Have a great night. I will see you all whenever we do this again. Thanks, everybody. Stay healthy.